Okay, welcome everybody. It's my great pleasure to welcome you all here to today's symposium on culture and longevity, which is sponsored by the IBSA Foundation. I mean, everybody seems to be interested in this topic, so I think you know why you're here. ETH is hosting this event together with IBSA for several reasons, of course, right? This is of course, a highly relevant topic to the society. We all know that our age is increasing, and with age you have, of course, the increase in certain disabilities, certain diseases, certain other kind of deficiencies, and the big question will be, how can we address this to keep the population healthy for a longer time? And this is clearly a research question. And on the other hand, you have the question, what does it mean if the population ages? What does it mean with regards to changed requirements, changes in architecture that may be required in the future to coordinate or to help stem these effects? And of course, here comes in the cultural aspects. We know that, of course, exercise, we'll get a very nice talk on this. We know that nutrition, we'll get another very nice talk on that, play a huge role in aging. But I think it's also clear that social aspects in life affect aging, and there will be talks on this. And I think that's quite a new field. And this is, again, why it makes it so interesting for the ETH. Because new technologies, especially in the imaging sector, or new technologies with tracking and other digital systems, and maybe even new digital technologies that encompass certain computational measures, can be implemented to really start to understand these relationships, and maybe they can be used again to affect what I mentioned in the beginning, changes in the aging society, and ultimately keep the society healthy. And this is the last aspect to it, and this is what I'm, as ETH Vice President, quite happy about, is the link to Ticino. The IBSA Foundation is located in Ticino, and as some of you might know, there was yesterday there was an event in Bellinzona, a very similar, so it's a partner event Bellinzona, now event at ETH Zurich. And for those of you who follow the medical landscape, you're aware that there's very strong ties between the area of Ticino and the ETH Zurich. We have jointly started a bachelor and master in medicine, and I think this is a chance to coordinate our efforts even more, not just on the research side, maybe also on the cultural side, and to promote research in longevity and add this angle of culture to it, and maybe thereby expand the field. And I don't want to keep you too long, right? You want to listen to the real science and not to me. So I will hand over to Silvia Misti, she's the head of the IBSA Foundation, and she will introduce you to the foundation. Thank you very much for coming, and enjoy this nice series of lectures.
Thanks, Christian. Good morning to everybody, and welcome to this important forum from IBSA Foundation. The forum of today promises to be really fascinating and education, so thanks Professor Wolfram for accepting this very nice collaboration, and we are very honored to be here in this institute. Since it has been my privilege to lead the IBSA Foundation for Scientific Research for 10 years, I'd like to give you a brief overview of our activities, all of which aim to reduce the gap between scientists and the general public. So just to don't take so much time for this introduction, we have a 1-minute video for all our activities. Thanks, Jacopo. Thanks. Given the relevance of aging in the world and also, of course, in the Swiss society, this forum aims to promote awareness about aging in both the scientific community and in the population. The biology of aging and disorders are the main topics of the forum, the first section. The speakers that we have invited will address the most recent discoveries in this field from molecular, cellular, and organismal angles, giving us a chance to learn and discuss new concepts and pressing issues. In the second section of the forum, after the coffee break, epidemiological, scientific, and psychological information will be combined to discuss the crucial role those social connections, culture, and arts play in determining our happiness, health, and longevity. So, as you can see from the program and from this special template of this forum, also today is already an experiment that we are trying to do. The link between culture and arts and the longer life expectancy and happiness will be also discussed. That's why, because we believe that culture and arts have a positive impact on people's health, so we'll have a talk on this. This is why in Ticino, IBSA Foundation, in collaboration with the city of Lugano, with the cultural division, we launched a culture and health project in 2020. The goal of this project is to demonstrate the positive impact of arts and culture, but using scientific approach and method. That's why we have promoted a university course on this subject and for medical students, and forums, clinical studies are ongoing. Also, I would like to tell you that we have opened a very nice and interesting website. This is cultureandhealth.ch, where you can find all the best practices, papers, publications that have been published in the world or organized with this goal, trying to demonstrate this role of culture and art on health. Luigi Di Corato, Director of Cultural Affairs of the City di Lugano, will be introduced to you in the second part to discuss about this. So just for concluding, this forum will also emphasize the need for interdisciplinary collaboration among health professionals, researchers, specialists from other fields, as you see from the program, so that aging and longevity medicine can be considered a real and established clinic and academic discipline. By working together, professionals from different areas can bring their different perspectives and experiences to the table, creating more effective approaches and solutions to aging-related issues. So from my side and from my team that is working very hard on all our events, good work. We'll be happy if the discussion of this forum is lively, so I hope that you will have a lot of questions to do after each talk and also at the end. And so we have time to do that. I will now leave the floor to Professor Andrea Alimonti. He will introduce himself, the forum, and then the speakers. Thanks for attention.
So thank you. Hi. Hello, everyone. So thank you very much for being here today. So it's a great pleasure to introduce the speaker of this today's section. My name is Andrea Limonti. I'm a full professor of oncology here at ETH and IDUSI in Lugano. So today I will moderate the section together with Christian Wolfram. So today I have the pleasure to introduce 4 speakers. Two speakers are Guido Kroemer and Professor Thomas Rando, are worldwide recognized for their contribution to the field of aging, cancer biology, stem cell biology, rejuvenation therapy. The other two speakers, Professor Schulz and Dr. Bon, are experts in psychosocial determinants that can affect longevity. So they will give a very interesting talk. So by saying that, let's start with introduction of the first speaker of today, Professor Guido Kroemer. So Guido is currently professor at the Faculty of Medicine of the University of Paris, Cité, director of the research team Metabolism and Cancer Immunity at the INSERM, and is also director of Metabolomics and Cell Biology platform of the Gustave Roussy Cancer Center, which is one leading cancer center in Europe. So to go through all the scientific contribution of Guido will take more than 30 minutes, but I just want to remember two important contributions that he has made in the field of science and cancer and aging. First contribution, I think, is the discovery of the program cell death. So he has really given a lot of push to this important topic. And the second is, of course, the discovery of an immunogenic cell death. He has recently also been becoming extremely famous in the field of aging for having written, together with other, an important review that summarizes all the hallmarks of aging. So for me, it's a curiosity of Guido. Is also one of the highest cited cell biologists around the world. So the title of Guido's talk is Hallmarks of Aging, Focus on Autophagy. Guido, the stage is yours. Thank you.
So I already gave the structure of my talk. The reason why I'm invited here is because we wrote 10 years ago a review on these hallmarks of aging that are already defined in a way, not in the details. We published a few years later the hallmarks of health, so the biological processes that allow the maintenance of health defined in a positive fashion instead of the absence of pathology. And at the beginning of this year, we published the update of the hallmarks of health that you see have become more numerous. And actually, it was a difficult task because during the 10 years that elapsed from the first version of the hallmarks of aging and the second version, there were as much paper published, more than 300,000, then were published on aging during the entire story of PubMed, maybe humanity, before. And so this means that the aging field is fastly evolving, and in a way it's a good news because science gives us the hope to intervene on one of the most severe problems that affects humanity. So here you see the graphic representation of the hallmarks of health. We identified 12 processes that, of course, are intertwined. It's a didactic representation of the aging process, through which I will walk you very quickly. So there are hallmarks that affect the stability of the genome and the epigenome, meaning that with aging, DNA damage and mutations accumulate, passing the time, also due to the reduction of DNA repair. And there are changes in the epigenome that usually define the identity of cells that becomes less than sharply defined and affects hence post-translational modifications of DNA, histones, RNA, and so on. We have hallmarks that are dealing with the turnover of proteins that has to be maintained, proteostasis, or the turnover of protein aggregates and organelles in the cytoplasmic cells that are handled by macroautophagy, which is disabled as we age. And so loss of proteostasis leads to the aggregation of peptides and misfolded proteins in inclusion bodies. and disabled macroautophagy causes to the failure to control pro-inflammatory molecules and hence unrestrained inflammasome activation and the accumulation of dysfunctional organelles, in particular mitochondria, which are the powerhouses of our cells. And there's, of course, some cellular damage and the potential initiation of oncogenesis through the disabled macroautophagy. There are biochemical hallmarks of aging that affect nutrient sensing and mitochondrial function. So as we age, the insulin growth factor receptor pathway, that is also important for diabetes, is overactivated, leading to a stimulation of the mTOR pathway and excessive anabolic signaling, again, excessive unrestrained inflammation, inhibition of autophagy, and mitochondrial dysfunction, which has different kinds of etiology, at the end leads to reduced cellular fitness, increase of inflammation due to the leaking out of DNA from dysfunctional mitochondria, and release of pro-apoptotic factors and excessive cell death. We have hallmarks that affect cellular functions, in particular cellular senescence and stem cell exhaustion. And you have to know that senescence is actually a physiological response to subclinical stress that is important, for instance, for wound healing. And it is the failure to eliminate senescent cells, for instance, by the immune system, by T cells, or the failure of endogenous programs to dissolve senescent cells by programmed cell death, that at the end cause aging and disease, or even cancer. We have hallmarks that are much more complex because they are supracellular, that affect intercellular communication systems, for instance, neuroendocrine circuitries, chronic inflammation, that is also called inflammaging, because it is a constant process that leads and accompanies the aging process, and dysbiosis, which is a disequilibrium state between our intestinal microbiota and the host that affects the so-called meta-organisms. And so excessive inflammation fuels progressive tissue deterioration and different types of dysbiosis that usually reduce bacterial diversity and lead to the production of toxic metabolites, accompany the aging process. And all these hallmarks actually can be intervened at experimentally in mice and sometimes in clinical studies to improve health and longevity. So these are the complicated biological hallmarks of aging, but the things are even more complex because they act through all the organismal strata of the organism, from molecules to organelles, cells, supracellular units, organs, organ systems, systemic circuitries that connect everything, and, as I already mentioned, the meta-organism, also called holobiont. And all these strata are actually also affected by the hallmarks of health that deteriorate over time, creating a highly multidimensional network of alterations that are communicated through the system and explain the aging phenotype. Now, there are some peculiarities because aging is usually accompanied at a given state by the manifestation of diseases, which can be, for instance, cardiovascular, neurodegenerative, and cancer, and there are differences. So, for instance, cardiovascular neurodegenerative diseases are affecting increasingly unfit cells and systems in an aging organism. So both are unfit, the unfit organ and cell and the unfit environment. But cancer is different because the cells that form a cancer are subjected to a Darwinian selection and getting ever fitter and selfisher in the unfit environment. And so if we compare the hallmarks of cardiovascular disease with those of aging, it is just a sub-ensemble of all the hallmarks of aging. When we look at the hallmarks of aging, the hallmarks of cancer that have been described by Doug Hanahan, there's not so much equivalence. So some of the hallmarks of aging, hallmarks of cancer are the same, and some are actually antagonistic or only partially comparable. So we have the so-called meta-hallmarks of aging and cancer that affect genomic instability, epigenetic alterations, chronic inflammation, and dysbiosis. But we have other alterations, so for instance, stem cell exhaustion and telomere attrition that are hallmarks of aging, but tend to render more difficult malignant transformation. And we have ambivalent hallmarks, such as disabled macroautophagy, that affect the fitness of the organism, but also the fitness of this cancer cell. So in some circumstances, this cancer cell needs autophagy to stay fit and hence has an ambivalent role in the process of transformation. And then there are tumor-promoting and metabolic commonalities among hallmarks of aging and cancer. So let us talk about starvation. You could call it hunger when it becomes unpleasant. And that's the reason that we have to invent caloric restriction mimetics that induce autophagy, which we retain as one of the best or most easily actionable entry points into the aging system for manipulation to accelerate or decelerate aging. And so what we try to achieve is to induce autophagy, which is slowing down with aging, to maintain the health and the fitness of the organism. We actually came into this because we worked on a different question. We asked ourselves what would be the mechanisms through which pharmacological, genetic, or nutritional manipulations, including caloric restriction, would increase health span and lifespan in model organisms, which are flies, nematodes, yeast, and mice. And it turned out that whenever such a manipulation works, it fails to do so if we inactivate essential autophagy genes, suggesting that autophagy is required, perhaps not as a unique phenomenon, but it is required for these interventions to work. And so often this occurs through the action of these caloric restriction mimetics that, if they are used in mice, actually have not just effects on longevity, but also on health span and immediate anti-obesity, anti-diabetic, organoprotective, and sometimes even oncopreventive effects. So what is autophagy? Just a brief review. This is a phenomenon that you can see by conventional light microscopy because cells become vacuolated and sequester in the cytoplasm structures that can be aggregated proteins invading pathogens, degenerating organelles in so-called autophagosomes, which are like the mouth of the cell that closes on the object that has to be eaten, because autophagy means etymologically self-eating. So the mouth then will fuse with the stomach of the cell, which is the lysosome, to create autolysosomes, and for the luminal digestion of the autophagic cargo. So macromolecules are converted into metabolites, the basic building blocks of future adaptive responses of the cells, and ATP is generated to provide energy. It is extremely complicated how autophagy is regulated, and I will not get to the details. I just mentioned the mTOR pathway as an autophagy inhibitory pathway. But it is easy to be measured because we can follow the subcellular localization of proteins from the LC3 family, which is usually all over the cell, in the cytoplasm, in the nucleus, everywhere, and distributes to autophagosomes and autolysosomes when autophagy is induced. So you get a punctate staining pattern when you follow LC3. And this is an example. So LC3 is marked with GFP. In the negative control, there are no puncta. With the pharmacological mTOR inhibitor taurine, you get puncta. And so using this as a screening system, a phenotypic screening system by microscopy, one can identify caloric restriction mimetics. And there are a few of those that we discovered to be autophagy inducers in our laboratory. For instance, resveratrol, spermidine, and some chalcones can delay aging and increase the lifespan of all sorts of model organisms. For spermidine, we even have epidemiological associations from nutritional studies that people who take during that lifetime a higher amount of spermidine have a lower all-cause mortality than people who don't take up enough spermidine. And these agents usually also have anti-obesity and anti-diabetic effects. Spermidine even slows down brain aging in flies and mice. We have caloric restrictions, including caloric restriction mimetics, including aspirin and nicotinamides that can delay the manifestation of oncogene or carcinogen-induced cancers. We have caloric restriction mimetics that improve the efficacy of cancer chemoimmunotherapy. There are other caloric restrictions that we can use in patients in phase 2 trials to show that the cystic fibrosis caused by the most frequent mutation in CFTR actually respond by a reduction of lung inflammation, and we know from mouse studies that this is mediated through the induction of autophagy, and we have effects of caloric restriction mimetics against cardiovascular disease. And all these changes that I list here are due to the induction of autophagy because pharmacological inhibition of autophagy or better genetic inhibition by knocking out autophagy-related genes abolishes the beneficial effects of these CRMs. Now, it's more or less classical pharmacology, although some of these agents are natural compounds, like spermidine, that is an endogenous metabolite, or nicotinamide. But still, they tend to be small molecules acting on intracellular targets, exactly like classical cancer drugs. And as I told you in my introduction during this slide disaster, we have these immune checkpoint inhibitors, which are monoclonal antibodies used against immunosuppressive extracellular proteins, such as PD-1 and PD-L1, that now are the most lucrative drugs for the pharmacological industry. This is an aside, but most importantly are the most efficient anti-cancer drugs that we have now in the clinics. And so the question that we asked ourselves is whether it would be possible to find autophagy checkpoint inhibitors, so antibodies that can utilize an extracellular target to get a pro-autophagic outcome. And so we started to speculate in the following way. Stress of any kind, endogenous or exogenous, including nutrient stress, can lead to an intercellular adaptive response that leads to cellular adaptation or not. And this will, in the case of adaptation, lead to cellular homeostasis and organismal homeostasis. If it does not occur, there's an irreversible change that leads to permanent cell cycle arrest in the form of senescence or to programmed cell death. And it is known from the literature that senescence and cell death leads to danger signaling in different forms, the senescent-associated secretory pathway and danger-associated molecular patterns that then signal to neighboring normal cells, or even through the distance to the entire organ systems and the entire organism, and allow for systemic adaptive responses to allow, hopefully, the reestablishment of organismal homeostasis. But what we did not know as we started our project was whether autophagy would lead to a sort of danger signaling. And so actually the whole project started with reading papers in unrelated fields. So there were these 3 papers that came out in 2010 showing that 3 different species of unicellular or facultatively multicellular fungi can release a protein called ACBP, acyl-CoA binding protein, if they are starved. So in response to neutron stress, these yeast species release this protein, which makes no sense. Why should a unicellular organism release a protein? And why should it do so in the context of stress, where it is just a waste of energy? So we wondered whether this protein might have a signaling function. And so it turned out that ACB-DBI is actually phylogenetically conserved throughout the entire eukaryotic radiation, but also found in some eubacteria. It is found in plants, in animals, in fungi. It is very similar in structure, short protein, smaller than 100 amino acids, and it comes in with 2 names, in mice and in humans. ACB stands for acyl coenzyme A binding protein when it is intracellular, and the very same protein has a different name, DBI for diazepam binding inhibitor, when it is extracellular. So the transition between these 2 pools is mediated by an atypical form of protein secretion which is autophagy dependent, but Golgi independent. And if you look at the amino acid sequence, it is practically identical between mice and humans. It is very similar for yeast, so we started to work on this protein first in yeast. And so we knocked it out and showed by biochemical tricks that autophagy is stimulated if we knock out ACBP-DBI from the system, and at the same time that the chronological lifespan of yeast was increased. So controls and black live about 5 to 6 days, but delta ACBP1 knockout yeast, which is the equivalent of ACB-DBI, live 50% longer. And this lifespan extension is lost if we knock out essential autophagy genes such as ATG5 or ATG7, meaning that we can conclude that longevity extension by removal of ACB-DBI is possible due to the stimulation of autophagy. And in addition, we found that the knockout of ACB-DBI reduced the capacity of yeast to form spores in conditions of starvation. So yeast has a particular life cycle, very primitive. It feeds until the food resource is exhausted, and then it forms spores to get into the air and fly until another food source is found. And by the way, this spore formation is also coupled to gametogenesis, to switch from diploid to haploid genomes. So if we knock out ACB-DB, we have less spore formation that we can rescue, at least partially, by adding recombinant yeast ACB-DBI to the system. And this rescue requires the pheromone receptors ST2. So it's a receptor-mediated process. And when we study then not only yeast, but also Drosophila, C. elegans on mice, we come up with a stereotyped pattern of 2 biological phenomena that are regulated by ACB-DBI. Starvation that causes its release from cells due to autophagy, the extracellular ACVDBI then will act on receptors that cause an adaptive increase in food intake. More mastication in worms, food-seeking behavior in mice, sporulation in yeast. And this is a homeostatic feedback loop because it removes the cause of ACVDBI release, which was starvation. But there's also a disease-amplifying feed-forward loop because when organisms age, mice or humans, the levels of ACVDBI rank up in the extracellular fluid due to an increased expression of the protein. It is also increasing in multiple inflammatory conditions and in obesity. Then the pool of extracellular ACVDBI that acts on its receptors inhibits autophagy. So inhibition of autophagy is useful in the case of the starvation response because you want to prevent the endogenous macromolecule resources to be eaten up. But in the case of disease, when you have to rejuvenate your cells and preserve them from stress, it is maladaptive. And this is actually a phenomenon that we see often in biological circuitries. It is called antagonistic pleiotropy. So the same process, roughly, that is useful in one situation can become maladaptive in another situation. And so the molecules that serve for life and youth are also the molecules that determine the aging process and are potentially pathogenic and even lethal. And so I will not go into detail. ACVDBI is within cells binding to medium and long-chain acyl-CoA esters and participates in lipid metabolism. It is released from cells upon stimulation of autophagy and stress death, and it acts, that's important, on the gamma-2 subunit of the heteropentameric GABA receptor. And it is through this action in mice, and presumably humans, that it induces appetite and inhibits autophagy. So we can intervene on these circuitries by neutralizing extracellular ACVDBI with antibodies. And we do this, we take out their action on their receptors, and we block increased food intake and autophagy inhibition, and instead we obtain inhibition of food intake and induction of autophagy. And of course, we take out the circuitry. So a few data from humans. In anorexia, ACVDBI goes down in the plasma. In obesity, panel B, it goes up. It correlates nicely with body mass index in panel D, and it correlates also with biochemical markers of prediabetes and liver damage in panels E and F. And in mice, we get the same results, and we can intervene on the system by injecting antibody against ACVDBI. And you see here by comparing weight gain per month, the black boxes in control and the blue boxes with the antibody, that you reduce weight gain induced by leptin mutation or weight gain induced by a Western-style diet. Now, if you look at tissues from humans in different pathological situations, ACVDBI goes up in multiple cases. The only case where it goes down is in anorexia, but it goes up in chronic inflammation, cardiovascular disease, hepatic and kidney injury. It goes up in cancer, certain types of cancer, neurodegeneration, diabetes, immune dysfunction, osteoarthritis, and as a general phenomenon, it accompanies increases in body mass index and aging, and both correlations that you find even in undiseased individuals are independent from each other statistically. So if you believe in the disease-amplifying role of ACVDBI, we should inhibit this protein by suitable monoclonal antibodies or by induction of autoantibodies, by auto-vaccination that breaks self-tolerance. Or we can intervene genetically by knocking out the ACVDBI gene, or by mutating the receptor, and then we can show genetically that the immune effects are on target. And what is remarkable here is that there are no side effects of the knockout in adult mice, and that the receptor effects are not mediated by an effect on the central nervous system, but are mediated on peripheral organs that express the GABA receptor. So when we knock out, when we take away the different ingredients of this pathway, instead of inhibition of autophagy, we obtain induction of autophagy because you remove the checkpoint. And so in mice, we can show that neutralization of ACB-DBI mediates effects on the central nervous system, neuroprotection, cardioprotection, liver protection, protection of the lung, and anti-obesity effects, and I will show some of them when they are linked to the aging process. So let us take the example of the liver. So we can protect the liver in this continuum that leads from health to non-alcoholic steatohepatitis NASH to fibrosis and cirrhosis by looking at different disease models that reflect each of the steps. And if we neutralize extracellular ACB-DBI, we induce autophagy, and we protect the liver against damage. For instance, damage induced by paracetamol, which is the most frequent cause of acute liver failure in the United States. You see that paracetamol induces these necrotic foci in red that if we combine APAP or paracetamol, APAP is a technical word, with anti-ACB-DBI in blue, we see less of these foci. And if we simultaneously inhibit autophagy with hydroxychloroquine, we lose the beneficial effects of the antibody, meaning that it was mediated through the induction of autophagy. And so we believe that the acute hepatoprotective effects of ACB-DBI neutralization depend on autophagy induction. And we can work through different models. For instance, we can look at the so-called methionine choline deficient diet, which is a rapid model for the induction of non-alcoholic steatohepatitis, NASH. And you see that in red, the toxic diet causes liver damage that has been quantified here, and the increase of transaminases such as ALT, which is a sign of hepatocyte death, and the knockout of ACB-DBI, the monoclonal antibody against ACB-DBI, the autoimmunization against ACB-DBI, or the mutation of the GABA receptor gamma subunit that abolishes binding of ACB-DBI to the receptor all have a similar disease-attenuating effect in this model. And we can look at the genetic profiles, and in these volcano plots you see that in this disease model, the neutralization of ACB-DBI causes to a downregulation of inflammatory markers, a downregulation of macrophage recruitment into the liver, a downregulation of fibrotic pathways, and a simultaneous upregulation of antioxidant enzymes. And so if you look at the entire spectrum of gene expression changes, so RCD is regular CHO diet, MCD, methionine choline deficient diet, induces NASH and induces these huge genetic variations that are basically abolished by neutralization of ACB-DBI. And now you can do the trans-species bioinformatic analysis and compare mouse data with patient data. So ask the question whether the genes that are upregulated by the MCD diet and downregulated by the treatment with anti-ACB-DBI antibodies, this segment of genes, is it also the type of genes that are overexpressed in NASH? And we did it for 10 different data sets, finding always the same result, that there's an overrepresentation statistically between mouse genes that are apparently pathogenic and human genes with a p-value that at the end is ridiculously small, close to 0. And we can look at different models, more realistic for people living in the developed world is Western-style diet with a little bit of liver damage that could be represented by alcohol or by CCl4. And you see that the signs of hepatosteatosis with these ballooning hepatocytes, or the signs of fibrosis in orange, that is a stain of collagen, are immensely impressive with this treatment that is strongly reversed or slowed down by the treatment with the anti-ACVB-DBI antibody. And now you can study senescence in these organs by staining the liver with an antibody against P21, which is a cyclin-dependent kinase inhibitor that is typically upregulated in cellular senescence. You see with the toxic diet that there are these brownish hepatocytes that appear in the tissue that are totally eradicated by the monoclonal antibody. And you can move to other organ systems. For instance, this is a model of accelerated lung aging induced by bleomycin instillations, accompanied by fibrosis, inflammation that is attenuated by intravenous injection of ACVB-DBI. Or can look at ischemia reperfusion damage as a model of myocardium infarction. That is, when you measure the necrotic volume, is in red the controls affecting roughly 60% of the myocardium and less, around 40% with the antibody against ACVB-DBI. And in this experiment, we also worked with hearts in which the ATG7 gene and the sensory autophagy gene had been selectively removed in cardiomyocytes. And here you see comparing the green and the brownish group that there's no effect anymore of ACVB-DBI, meaning that these beneficial effects are again mediated through the induction of autophagy. Back to humans. We took advantage of a cohort from southern France of apparently healthy, mostly women that were interested in prediabetes prevention and found that those patients that after blood sampling in the forthcoming 9 years would develop a cardiovascular event. These were 50 among 600 persons that we matched for age and body mass index with apparently healthy controls that stayed healthy later on, and found that future cardiovascular disease or cancer was associated with elevated ACVB-DBI levels, as if this factor would not only correlate with chronological aging, but also with biological aging that is measured by the future disease. And we validated this in another cohort to be sure about the result that there's an association between cardiovascular disease, future cardiovascular disease, and ACVB-DBI elevations in the plasma. As an interesting side observation, we found that there's usually a correlation between age and ACVB-DBI levels in people who stay healthy. But in those who will develop future cancer, future cardiovascular disease, this correlation is lost, as if there was some regulation circuitry that goes astray. And this is also found in meta-analysis across different cohorts, that in future or present disease, the correlation between ACVB-DBI and age, or the correlation between ACVB-DBI and BMI is lost. And when we look just at cardiovascular parameters, we see that typical biochemical risk factors such as increased total cholesterol or increased triglycerides correlate with ACVB-DBI, and some of these correlations withstand multivariate analysis and are independent from age and BMI, as is true for total cholesterol, a positive correlation between ACVB-DBI and the laboratory parameter, and HDL cholesterol, which is the so-called good cholesterol, which is cardioprotective, whereas an anti-correlation between ACV-DBI and the laboratory parameter. And then we can move back into mice and ask whether ACV-DBI neutralization might delay cardiac aging in a model of accelerated aging induced by anthracyclines, which, as you know, in breast cancer chemotherapy are inducing cardiac failure as a side effect. And so if we look at the non-invasive hemodynamic parameters, the left ventricular end diastolic volume, normalized to body surface area that goes up with doxorubicin treatment as a sign of cardiac failure, is normalized by neutralization of ACV-DBI. And again, we can look at the histology of the cardiac muscle, seeing these senescent P21 positive cardiomyocytes induced by doxorubicin treatment in brownish color that vanish if we treat the mice with antibodies against ACV-DBI. And then we can actually look also in other organs, such as the kidney, and find that podocytes in glomeruli are induced to become senescent with doxorubicin, but not so anymore in the presence of the antibody, and interstitial kidney peritubular capillaries behave in a similar fashion. So we can conclude from this overall that aging is a complex process governed by multiple genetic, protein affecting, cellular phenomenon affecting biochemical and supracellular hallmarks that can be targeted in part by acting on macroautophagy, reactivating this phenomenon that's usually disabled with progressive aging. And when we reactivate autophagy, we also repair other aspects of the system. For instance, we inhibit mTORC signaling, which I didn't show. We abolish cellular senescence, and we counteract and inhibit chronic inflammation. And there's probably other things to discover in the effects of autophagy on the other hallmarks of aging. And the other interesting aspect of my talk is that you can walk through the phylogenetic ladder and perform lifespan extension experiments in yeast to find biomarkers of future disease in humans and intervene on mice to reverse organ-specific aging and perhaps the aging phenotype at the whole, which we still have to confirm. With this, I would like to mention some people from my laboratory, the many, many collaborators that have been contributing to this project, because you remember that science is always a collaborative adventure. Thank you for your attention. Questions?
Christian?
Thank you very much. This was very interesting. I actually have two questions. You showed ACVP as the autophagy regulator, but this is, of course, an intracellular effect. But then in the patients, you measured ACVP in the serum, if I'm not mistaken.
It is the extracellular ACV-DBI, the secreted version of ACV-DBI, that acts on receptors of cells to inhibit autophagy. And if we take out all pools of ACV-DBI by knockout, so we then remove both intracellular and extracellular ACV-DBI, or if we just add an antibody that neutralizes obviously only extracellular ACV-DBI, we get a similar autophagy stimulating effect.
And how does this regulate autophagy? I mean, this is a acyl-CoA binding protein, right? Or it's a part of it at least.
Yeah, but the acyl-CoA binding effect of ACBDY is not important for autophagy regulation because we can mutate the binding site to acyl-CoA, and we will not lose the autophagy regulatory function of ACBDBI. Conversely, we can mutate the receptor of ACBDBI, and then we lose the autophagy modulatory function of the ligand.
Okay.
So we believe that for autophagy, it is mostly the extracellular pool of ACBDBI that is relevant, and that is actually then accessible to intervention by antibodies. And that's also the reason why I believe that measuring extracellular ACBDBI in body fluids, such as the plasma, is relevant for studying indirectly the autophagy regulation in mice or humans.
Thank you.
I think there are two questions there. Okay.
Thank you very much for your talk. I have a general question about metabolism. How advantageous is it to slow down aging and get into a starvation mode while reducing metabolism? Because there are also disadvantages of slowing down a metabolism, right?
So do you ask about cell type specific aging phenotypes?
No, it's a very general question. You mentioned that aging could be slowed down by starving a cell.
Yeah.
But at the same time, the metabolic energy origin is really within. It will be reduced as well. The mitochondrial function will just go down.
It's always a question of dosing. So in pharmacology, like in physiology, too much of a good thing is at the end bad. So caloric restriction is a good thing within limits. Limits that can be defined in different ways. So you can give a mouse every day just 70% of the amount of food that he usually would eat. Or you can restrict the time a mouse has access to food to, instead of 24 hours, just 6 hours. And of course, if you go beyond a certain threshold of caloric supply, then the mouse will die from starvation. So you have to find the right dose to get the positive outcome. But it's true for drug interventions as well. So if you give a drug that is beyond the threshold, it will induce off-target effects and toxicity.
Thank you.
There is a question. Collins, I was first, and after another one.
Thank you. Wonderful talk. Really exciting to have, this checkpoint inhibition for autophagy. Opens up a door to a lot of different interventions. So I would like to pick your brain. You've been surely thinking about combination, right? So with what intervention would you combine now to activate autophagy? Do you have any next steps?
Well, so that's a perfect question. So it will also occupy for the next century because it's also a complex question. So which types of anti-aging interventions should be combined to get additive or maybe even synergistic effects? And so roughly speaking and very theoretically, I would not combine different autophagy inducers among each other because they act in the same pathway. So beyond a certain threshold, presumably they would also become toxic, and it makes intuitively no sense, although it should be tested because you never know in biology. But of course, it would be much more interesting to combine interventions that act on different hallmarks, hoping to define those that are practical and non-toxic in their combination and mediate these additive or hyper-additive effects that we dream of.
Hi, thank you. Very inspiring talk. I have two small questions. One is caloric restriction reduces inflammaging. Do you think that that is entirely explained by autophagy, or is there something more to it? And the second question is related to the previous one in combining interventions. If one intervention, a particular dose that you can give body-wide, clearly has different effects on different organs, and the appropriate dose differs between organs. Do you have any thoughts about how to optimize the administration so that you basically try to get sort of appropriate effects body-wise?
Okay, so the first question was on inflammation and its inhibition by autophagy. So there's a literature that suggests that you can induce autophagy to inhibit inflammation and to, at the same time, stimulate immune responses, which appears a little bit contradictory because often people put the inflammation and the immune response in the same category, but this is complex. There are different cell types in different situations, constellations. And so when you induce autophagy experimentally in mice, the inflammatory parameters that you can measure are reduced. And this is shown genetically. So when you mutate BCL2 in a way that it cannot inhibit autophagy anymore, you get a hyper-autophagic phenotype in mice that is coupled to increased longevity and reduced inflammation. And you get the same if you feed mice with spermidine through starting at a middle age or even at an old age. You extend lifespan and you reduce inflammation in different organs, including in the heart. So this, there may be a sort of hierarchy that among the hallmarks that explain that autophagy is one of the possible interventions on inflammation. It's certainly not the only one. The second question was dealing with, if I understood the acoustics was not very well, with chronobiology, dormancy of which.
And that could explain perhaps why the effects are not optimal, because you would need to somehow...
Yeah, that's an excellent point. So, for instance, in the liver, we have, it's actually all organs are similar constructed. You have these minimum supracellular units, which are parenchymal cells held together by fibroblasts and that are under sort of primitive immunosurveillance by tissue-specific macrophages in the liver. These are, the macrophages are Kupfer cells, and the relevant fibrocytes are stellate cells. And so it's only one of the cell types that express the GABA receptor gamma-2 subunit, which is the hepatocyte. So one could imagine that intervening on the other cell types with other drugs that have a stellate-specific effect, for instance, would give us combinatorial and positive effects.
Thank you. I think that we are running out of time, so thank you very much, Guido. You can ask questions later on at the coffee break. So it's a pleasure for me to introduce the second speaker of this symposium, Professor Thomas Rando. Professor Rando is the director of the Broad Stem Cell Research Center at UCLA in the US, where he's also professor of neurology and molecular cell and developmental biology. Professor Rando has made essential contribution to the field of biology of aging and the stem cell, having pioneered the use of heterochronic parabiosis in the study of molecular, cellular, and organismal aging. He has published several papers. He has been awarded with several recognition. He's also a member of the National Academy of Medicine and American Academy of Art and Science. So the title of this talk today is Stem Cell Aging and Rejuvenation, an Epigenetic Journey. Thank you, Thomas.
Thank you very much, Andrea, and thank you, Sylvia, again, for this invitation to participate today in this forum, yesterday in Bellinzona and today in Zurich. So, as I mentioned yesterday, it's always hard to follow Professor Kroemer, but I had the advantage of knowing what he was going to talk about, and actually changed my talk today. Yesterday in Bellinzona, I talked about aging and the influence of exercise, which is very interesting to us and to a lot of people. And in part related to what Professor Kroemer talked about, I decided today to talk about another aspect of an intervention that is a lifestyle intervention that can influence aging, and that's diet. And so you heard about caloric restriction, and I'll end up talking about that. And so my talk is a little different than what my title says. So can I have my first slide, please? Or do I do that? I do, sorry. Okay, so we tend to think of the biology of aging very much through the lens of stem cells. And so we study how stem cells age, what the implication of those changes are for tissues throughout the body, both in terms of homeostasis of the tissue, just maintenance, as well as tissue repair and tissue regeneration. And this is just a figure from a review that I co-wrote with my colleagues, Anne Brunet and Peggy Goodell, with whom we study stem cell aging across the body. And this just illustrates that throughout the body there are stem cells. And they are participating often in maintaining tissues and repairing tissues, some of which, depending on this color code here, some of the tissues turn over all the time, like skin and the intestine of the gut and blood. You're always making new cells. So those stem cells are very active. And then there are tissues that turn over very little, and yet they have a remarkable regenerative potential. And that's the case for muscle, which is what we mostly study, stem cells and skeletal muscle, which itself is a tissue that really does not turn over very much normally. Just as you're sitting, as you're walking, as you're working, if you're not weightlifting, if you're not engaging in kind of activities that would damage your muscle, your tissue doesn't turn over, and the stem cells are very quiet. And yet, this same tissue has a remarkable ability to regenerate in response to injury, and the cell that's responsible for that injury is shown here in this little sort of lavender cell here, which is called a satellite cell or a muscle stem cell. So each of these structures seen here, these large structures, are the so-called myofibers of the muscle. They're multinucleated cells. They're the cells that contract, the cells that make you move. But these little stem cells, they sit here in the muscle. They're very rare, and they sit in a quiescent or dormant state for weeks, months, perhaps years, without undergoing cell division. So they're professionally dormant cells. And yet, despite that dormancy, they're responsible for the regenerative ability of muscle, and that's illustrated in this slide. So again, here's a normal muscle seen in cross-section under a microscope. Here's a couple of days after a total injury to that muscle with a toxin, so that structure breaks down. You see a lot of inflammation, and those very few stem cells, they transition from this dormant quiescent state to this incredibly active proliferative state. And they generate so much tissue that within a week, you can see they basically have reformed a muscle, essentially by recapitulating developmental programs, the way muscle develops in embryogenesis. And then over the course of the next couple of weeks, that tissue grows and restores itself. So there really is this incredible ability of these very few cells to make a whole new tissue. So what happens as we age? So one of the things that we know, just phenotypically, is that as we age, we repair our tissues less well. And that's really across the body. So if you injure your skin as a young person, it heals very quickly. There's no scar formation, bones heal very quickly, and yet as we get older and older, we start to see an impairment of these regenerative processes. And we basically end up with impairment of regeneration and impairment and in development, sometimes of fibrotic injuries. So these quiescent stem cells have two modes, or these stem cells have two modes. One is this quiescent state, which can sort of undergo what we call chronological aging, so just living in the body throughout time, just like another cell that doesn't divide, like a neuron. And yet when it undergoes a lot of this division, it undergoes what's called replicative aging. But what we're interested really in is these quiescent cells, as the organism age, as a person ages, what happens to these cells over time? And what we see is, as I mentioned, you see this decline in regenerative capacity of muscle due, at least in part, to the aging of these quiescent stem cells, and I'll talk about what we mean by that. So here's an image of a young muscle from a mouse. Here's a few weeks after a single injury. Here's a few weeks after a sequence of injuries. You get perfectly good regeneration. But in an older mouse, even after a single injury, you start to see areas of unrepaired muscle, this whitish area, and then after a second injury, you see even a more impaired regeneration. So the stem cells are clearly different at the beginning of this injury, leading to this impairment of this regeneration in older mice. So what's wrong with these cells? And we've studied this a lot. One of the things that happens that we found that with age, these cells become less resilient. So what does that mean? So in this case, this is just a way of measuring basically cell death. And in the gray is the young animals, and the red is the old animals, just showing that the stem cells from old animals are much more likely to die in response to any kind of stress. So they just are less resilient, just as older people become less resilient. These cells become less resilient. And in fact, what's interesting about these cells is that they undergo a form of cell death called mitotic catastrophe. And what that is that when they activate out of this quiescent state, they try and divide, they die during the process of cell division. And this is just a very simple experiment where you can't really see this, but each of these graphs is sort of a stacking up of lines of individual cells that we watched under the microscope when we asked, when does it divide and when does it die? And what you can see is the timing of cell division here really coincides, this is hard to use, with the timing of cell death. And so these cells don't just die when in the muscle, they don't just die when we put them in culture, they die when they try to divide. And this process of mitotic catastrophe has these very characteristic features that we can see. And so that's the loss of resilience that we see in these cells. So as we study cellular aging, one of our fundamental questions is always, is there anything we can do about it? Can we make this better? Is there a way that we can intervene to, in this case, protect the cells or enhance the ability of an old muscle to regenerate, to make it regenerate like a young muscle? And this is what Andrea referred to, the work we did, which is now starting about 20 years ago. We use this technique called parabiosis, whereby animals are surgically connected and they develop a single shared circulatory system. So the tissues of one animal are exposed to the circulating environment of the other animal. And so we have these control parabiotic pairs called isochronic pairs, so a young mouse connected to a young mouse, and the control old mouse connected to if I do it here, the old mouse connected to the old mouse, and in the middle you see the experimental mice, which are the heterochronic pairs. So there's a young mouse connected to an old mouse. They are left this way for weeks or months, and so during that time, the tissues of the old mouse are now exposed to the circulating environment of the young animal. So whatever might be positive effects from the young blood are now in the old animal, and conversely, anything that might be inhibitory from the old blood is now exposed, or the young tissues exposed to that. So what we found is that if we took those heterochronic pairs and we injured the muscle of the old mouse, it regenerates just as effectively as a young mouse. So the young tissues regenerate very well. The old tissue, this is basically a scar formation, but the old partner in these heterochronic pairs regenerate just as well as a young animal. So in that sense, the regenerative process has been restored back toward a youthful level, so a real rejuvenation. And we looked at the molecular features of the stem cells, and they have acquired a more youthful phenotype. And we looked at this in muscle, we looked in liver, and then a few years later we looked at this in brain with our colleague Tony Weiss-Corey. And then over the last decade, many investigators have used this technique of heterochronic parabiosis to show that old tissues can be rejuvenated. And again, I'll talk about what we mean by rejuvenation, because it's a word we like to use in quotes. It's a loaded word, but I'll tell you why we use it, what we think it means, and what the limitations of that are. So, is this really rejuvenation? And the reason why, I think one reason we can't answer that definitively is we really do not have what I would consider a molecular definition of cellular age, to say, this is the way we will define age by this set of molecules, young, middle-aged, old. We have a lot of features, but there is no agreed-upon definition. So we measure these features, we measure cell function. They can go from looking like old cells to looking like young cells, and it looks like a rejuvenation, but I think that issue, that definition is still, the jury is still out on that. But this is one way we think about this. And this is from a fanciful figure from a review I wrote with my colleague Howard Chang at Stanford now more than 10 years ago. And we modeled this kind of idea of the cellular phenotype as, so the genome is illustrated kind of as the film through which light is passed, through an epigenome, which is the lens, to produce an image, which is the phenotype, so how a cell functions. Now, with age, one of the things we know for sure, and I would argue is probably the real driver of aging, is genomic instability. So you get DNA damage, you get DNA mutations, all kinds of bad things happen to the DNA that are not repaired, and that accumulates with time. Now, at the same time, like Professor Kroemer described, there are a lot of things that are happening in the cell, like abnormalities of protein turnover, different marks in the chromatin, the proteins that surround the DNA, that are also changing. So not only is there a degradation of the DNA, the genome, but there's a degradation of the epigenome. And then in response to these interventions and others that I'll talk about, but like this heterochronic parabiosis, we're not repairing DNA, we're not restoring the genome, but what we think we're doing is we're rejuvenating the epigenome. So now that light is passed through a clearer lens, can't get that to work. Anyway, producing a more youthful phenotype. So it's an epigenetic rejuvenation, which is how we think of this, not a genomic rejuvenation. But I think to think about this, it's helpful to think about this from the context of other experimental paradigms and natural paradigms in which rejuvenation certainly occurs. And I'll try and convince you that this is true rejuvenation and how we think of this as potentially relating to our interventions. So most people in the room, many of the scientists in the room, would be very familiar with this idea of induced pluripotent stem cells. So what this paradigm is you take a differentiated cell, a blood cell, a fibroblast from the skin, and you modify that cell with these transcription factors called Yamanaka factors to produce these cells that are highly pluripotent, meaning they've lost all their differentiated characteristics and they're now pluripotent, able to form an entire new organism. So these pluripotent cells can then become sort of any cell in the body, including an entire organism. So they go through this process of de-differentiation, and then they can be redifferentiated. And that's the paradigm of pluripotent stem cells, the paradigm of iPS cells, and why that's such a powerful technique for understanding biology and for therapeutics, because you can use these cells to make all kinds of cells in the body, so all kinds of cellular therapy potentially. Well, I would say that there's another equally important process that's going on that gets a lot less attention, and that is whatever cell you take, that you start with, that cell has some age. It's come from a human or a mouse that is some age. It's not a newborn. And so what happens when you generate these pluripotent cells, you erase all that aging mark. So you reset the aging clock from whatever it was back to zero in this process of reprogramming. And then when you differentiate these cells, again, they acquire an age. They then go through this process of cellular aging. If they're in an organism, they age like any other cell in the body. So this is a true rejuvenation, a resetting of an aging clock from some age to zero, whatever that is. So cloning, somatic cell nuclear transfer, is exactly the same. So Dolly was the first mammal cloned from an adult cell. So the same thing, an adult cell, and the nucleus of an adult cell was taken, put into an embryo, put into a blastocyst, and that nucleus, which cloned Dolly, went from whatever age the donor was back to zero. So that embryo developed, Dolly developed, and all the cloned mammals have now undergone that same kind of reprogramming of age. Start with age, go back to zero, and generate an organism. And I would say that there's nothing more compelling to me than this image. So this is an image of a fertilized egg. So think of this again in this process. In this case, say in humans, you take two highly complex, highly differentiated cells, a sperm and an egg, very complex cells that are at least decades old. You put them together, and you have a cell that has an age of zero. It has to be true. If that were not true, we would pass on our age to our progeny, and every species would be extinct by the acquisition of age through every generation. Clearly, that does not happen. The very nature of life is based on the ability to erase the age of sperm and egg, to make a fertilized egg. So the age of that cell is 0. So rejuvenation is so fundamental to what we understand about life that we take it for granted. But this is true, and the question is, are the things that we as scientists doing actually doing anything like this in terms of rejuvenation? So one of the characteristics of these examples I've given you is this rejuvenation that occurs are all accompanied by dedifferentiation. So all of them, the cells that you start with, lose their features. They become completely undifferentiated cells. Whereas the kind of interventions that we're doing, like parabiosis, the cells are remaining as liver cells or muscle cells, but they seem to be younger. So the question we had way back when is, can you uncouple these? Can you reprogram the age of a cell without reprogramming the differentiation features of a cell? And that would be, to us, that would be kind of the holy grail of rejuvenation interventions, is to just modify the age and not modify the nature of the cell itself. So Juan Carlos Belmonte beat us to the punch on this, and he published his paper in 2016 in which they generated a mouse using these same Yamanaka factors, the one that make iPS cells. They put it into a mouse in a way that they could turn it on when they wanted to. And they gave these short pulses of Yamanaka factors, turning them on in cells, and what they showed, what they demonstrated was what looked to be rejuvenation of cells both in the culture and in the animal without dedifferentiation. So they didn't make pluripotent cells, they just made the old cells younger. So this is exactly what we had sort of proposed. We were working on this, came out several years later, using not the Yamanaka factor genes, but in fact the transcripts. And again, we gave cells, we gave animals these Yamanaka factors transiently, and we also saw a rejuvenation without a dedifferentiation. So these are, to us, kind of, this is the idea of reprogramming age that I think is really powerful. And yet still there's a lot to be known about what drives this, how it works, and how the cell responds. But what I'd like to talk about now is, we can use parabiosis in the laboratory. We can give animals Yamanaka factors, but we're not going to do this to humans. We're not going to give humans genes. We're not going to parabiose humans. So are there interventions that we can do, that humans can do, that could at least begin to mimic the kind of rejuvenation that we're trying to do experimentally? And in the aging field, the two big things to study are diet and exercise. And the beauty of that is that, again, like Professor Kroemer said, we've spent billions and billions of dollars studying aging, and pretty much it's what your mother told you: eat less and get some exercise. Everything comes down pretty much to the pathways that are regulated by diet and exercise. So yesterday I talked about exercise, and today I thought I would talk about diet. And I'm going to summarize, yesterday we talked about how exercise in a mouse can actually make an old cell appear younger. Again, we were looking at muscle stem cells, we looked at some of the pathways that are regulated, but exercise in the form of voluntary wheel running really could restore youthful properties to old cells. And the other thing I talked about yesterday that I think is important is the notion of muscle as an endocrine tissue. So when you exercise, not only do your muscles contract and get stronger and bigger, but they secrete things. They really are endocrine organs. And the things that are secreted, the factors that are secreted, at least proteins and probably other things, have these huge beneficial effects to tissues throughout the body. So that's why we think, when we exercise, people who exercise have lower rates of cardiovascular disease, have improvement in cognitive health, really have improvement in function of fat, so in bone repair. So really muscle is predominantly an endocrine tissue in this setting, and secreting factors that are having pro-health effects. So what about diet? So caloric restriction. I mean, like Professor Kroemer talked about caloric restriction. So this really is, this is the granddaddy of interventions for influencing the aging process. And this was the original paper by Clive McKay, in which he took rats, he put them on a caloric-restricted diet, fed them very little, and these are life curves. So these are the control rats. This is how long they lived. On average, about 600 days. These calorically restricted rats lived a lot longer. Okay, so a lot longer. And then this has been followed up in many, many species, as Professor Kroemer mentioned, and this is just a more recent example, but still many decades old, from mice. So this is the green, as you're seeing, the control animals that are fed what's called ad libitum, eat as much as you want, which is how we normally do this in the lab. And then blue is 25% caloric restriction, 55 and 65%. So this is a huge amount of caloric restriction. If you think about reducing your own calorie intake by 65%, that's a lot of hunger. But look at the effect on lifespan. So it really has a profound effect, and this has been demonstrated in just a wide variety of species. But an interesting thing still is, in my opinion anyway, is what do we compare this to? Because we compare almost always these interventions of caloric restriction to animals that are fed ad lib. And there's a flip side to being fed ad lib, and that is they get fat. So, I'll come back to that. What is the right control group for when you calorically restrict an animal? So this has been studied even in primates, so in rhesus monkeys. And there were 2 studies, one done at the NIA and one done at the University of Wisconsin, and they actually came to the opposite conclusions. One said that caloric restriction extended the lifespan, health span of primates. The other said they didn't, but they've now gotten together and they've looked at what they did differently. And the consensus is that there was a positive effect on lifespan extension in primates in a very controlled situation. And if you want, you can join the Caloric Restriction Society. You can calorically restrict. And of course, you'll never know if it's going to make you live longer, but people who do this firmly believe that they are improving the health of their cells in their body by calorically restricting for their whole lives. Not so fun, but it might work. Okay, so we've gone to this question of can caloric restriction, this is the intervention, the dietary intervention, does this improve the function of old stem cells and old muscle stem cells? Well, we haven't actually done this, but a lot of people have done these kinds of experiments. And this is just a paper from a colleague of ours. And I think it illustrates how complicated it is to do caloric restriction and draw these conclusions. So they did caloric restriction in 2 strains of mice. The mice are genetically identical, used exactly the same conditions, and they got very different results depending on the strain, depending on the age. So sometimes it worked, sometimes it didn't. And obviously, when you do caloric restriction, it depends on what you give the mice to eat. How many calories, what's in those calories, that makes a huge difference. So it's a very complicated paradigm, I think, to generalize about. So we've gone to an easier approach, which is fasting, because of course there's no food, there's no variable, it's just no food. And so we've basically asked this question: does fasting as a form of dietary intervention have a beneficial effect on and actually, a couple of the questions that were asked, I'm going to talk, I think, about things that relate to some of the questions that were asked Professor Kroemer. Does fasting enhance the function of stem cells and their role in tissue repair? And so this was work pioneered by a postdoc in the lab, Dan Benjamin, and a graduate student, Peter Both. And this is the way we did this. We just fasted mice for different periods of time, either a day, 2 days, or 2.5 days, which is a very long time for a mouse to be without food. And then we injure the muscle, but we refeed them. So the regeneration is occurring not during the time of metabolic stress. They're actually being refed. But in fact, what we found is fasting, however long we did it, made things worse. Someone was sort of asking about that question. It made muscle regeneration worse. So all the things that we think about fasting as being beneficial to health, when we do these experiments looking at stem cell function and tissue repair, it really impairs the function. So, but is there any benefit of fasting in some ways that is not reflected by our assays of tissue repair? And in fact, there is absolutely a benefit, and that is it makes muscle stem cells and aged muscle stem cells more resilient. So even though the regeneration process, all the features of the stem cells to proliferate, to make new muscle, are impaired, the cells sitting in this quiescent state are far more resilient, which is what you'd want them to be during this time of extreme stress. They're far more resilient to every stress that we've imposed upon them. So the cells really hunker down in response to fasting. And so I love this quote because, I'm not an evolutionary biologist, but everything I do in the field of aging makes us ask questions about evolution. And this, to me, as we started to think about this makes sense evolutionarily because in evolutionary terms, everything is a trade-off between survival and reproduction for propagation of the species. And when there's lots of food, animals reproduce. When the conditions in the environment get stressful, When food is not available, animals go into survival mode. And I think what we're looking at here is a cellular microcosm of that evolutionary example. So this kind of chronological aging, and especially when there's fasting that occurs during chronological aging, the cells go into this high survival, high resilient mode. And when they're not fasting, when there's food available, or they need to generate tissue, they go into this highly reproductive phase. So sort of analogous to species, there's a survival mode and a reproductive mode. And again, there are just so many good examples of species that reallocate their physiological resources depending upon the environment they live in. So in the setting of cold winters, in the absence of food, bears hibernate, they reduce their metabolic rate by 25%, and they go through this phase where they're obviously not reproducing, but they're in a profound survival mode. Arctic ground squirrel, they can live for 6 or 7 months in a hibernation state. They can survive temperatures almost to freezing by changing the composition of their blood. So again, this profound physiological adaptation. There are amphibians like the wood frog that can actually survive sub-zero temperatures with some of their bodily fluids actually frozen. The red flat-barked beetle has this really strange adaptation that in response to temperatures as low as 50 degrees below zero, they change their blood in a process called vitrification, so the blood is not even a liquid, it's a semi-solid, and they can survive freezing for months and months and months. Now, this is another good, fun example. So this is just in the plant. There are a million examples, obviously, in the plant world. So these are seeds that were excavated from a site in the Dead Sea that were found to be thousands of years old. They were put on a shelf, and then one day, someone decided to plant them, and they planted them, and they generated date palms. So again, this is just this highly survival mode, not reproductive mode, but as soon as they're given water and food, they can then produce. So, okay, so coming back to stem cells, so there's really this idea of a microcosm of this balance between survival and reproduction, or resilience and regenerative potential. So what I want to just finish on is, how does it work? So we fast the mice, and the cells go into this survival mode. How does it work? What is fasting doing? So one of the things that happens, or a prominent physiologic things that happens when mice fast, is we produce ketone bodies, so we become ketotic. And these ketone bodies circulate from the liver, they go into the blood, and basically go to cells throughout the body, and they serve as an endogenous food source or energy source in the TCA cycle. So this is the way cells survive in the absence of any exogenous food. And so what we asked was, if we don't fast the mice, but if instead we did one of two things, and I'll show one of these, we put them on a ketogenic diet, so they become ketotic, but they're not fasting, or we give them a normal diet, where we just give them ketone bodies. And the primary ketone body is beta-hydroxybutyrate. And what we find is that ketosis, whether by fasting, a ketogenic diet, or BHB, it basically protects cells, it makes them more resilient. And so how does this work? What is the mechanism by which these ketone bodies work? Well, so our first guess was, again, these ketone bodies are converted from beta-hydroxybutyrate to acetyl-CoA through this enzyme OXCT1. That's just the rate-limiting enzyme in the conversion. So we knocked out OXCT1 in these stem cells, assuming if we knock out this enzyme, it'll obliterate the ability of this metabolite to promote cellular resilience. Well, it had no effect. So even without being able to be used as an energy source, this metabolite enhances cellular resilience just as well with this enzyme or without. So how might that work? Well, it turns out our colleague Eric Verdin had published 10 years ago this evidence that beta-hydroxybutyrate functions in a non-metabolic way in the cell by blocking these enzymes, histone deacetylases. And so we just tested whether blocking histone deacetylases, which if the ketone bodies are high, they're blocking these enzymes. What if we just block these enzymes pharmacologically? What we found is that using this generic histone deacetylase inhibitor, we could recapitulate the effects of fasting. So just by blocking these enzymes, we see the same thing. And if we eliminated these enzymes, it obviated the ability of beta-hydroxybutyrate to generate cellular resilience. So clearly or not clearly, but I think this is evidence in support of the idea that these metabolites are acting through these enzymes. And so these are enzymes that deacetylate proteins. What are the proteins that are deacetylated by these enzymes that might be playing a key role downstream of the histone deacetylases? So these are some of our favorite proteins, but I'm going to talk about one that we found, and that's P53. So P53, when we fast the animals or we give them ketone bodies, we see a hyperacetylation of P53, which would be in that pathway between the metabolite, the histone deacetylase, and this protein itself. And if we give a pharmacological stabilizer of p53, which mimics the effect of hyperacetylation, we basically see that by this drug Nutlin-3, we see the same effect of fasting, HDAC inhibition, or p53 stabilization in terms of promoting this kind of cellular resilience. Okay, so just a couple of more pieces of data for the aficionados in the audience. So if we knock out p53 in these cells, the metabolite can no longer promote resilience. And if we use a mouse in which the p53 protein itself is genetically modified to look like a hyperacetylated protein, it produces the same effect as ketosis. Okay, so this is the model. Just getting back to the big picture. In response to fasting, these ketone bodies, beta-hydroxybutyrate, are produced in the cell. They're used for energy, but they're also used for something else. So they go into the cell, they block these histone deacetylases, and they turn on this p53 signature that promotes cellular resilience. And again, in an evolutionary sense, the thing I like about this is evolution has adapted or adopted this metabolite, which clearly is important for just survival and energy metabolism. It's adopted to use it to promote cellular resilience in the setting of nutrient deprivation. So the same metabolite doing two very different things, and one of them being this enhancement of resilience. So just to end, to come back to the question of rejuvenation, are we with diet, like we may be with exercise, like we may be with parabiosis, are we really inducing a reprogramming of the cell from an old state to a young state? I don't know. It looks like a young cell, it behaves like a young cell, it looks and tastes like a young cell, but I think until we really can define cellular age molecularly, it will always be this kind of phenotypic rejuvenation. So again, always in quotes, but there's a lot of, I think, interesting biology here. There's a lot of interesting potential therapeutics here. I mean, there are entire companies formed now on trying to reprogram cellular age, and so we'll see how successful. It'll be a tough approach, in my opinion, but we'll see. We'll see. Aging is a field that changes very dramatically, very rapidly. So just to finish, I just want to acknowledge, we had a lot of people work on these projects over the years. I've mentioned a few of our collaborators on the specific project, and of course I want to acknowledge the funding sources that have really allowed us to go from these very, very early simple findings down to these molecular pathways that I think are really informing us about what the nature of aging might be and the nature of interventions to slow, arrest, and maybe even reverse some of these aging processes. So I'll stop there and I'll be happy to take any questions.
Thank you, Thomas. Let me see if there is a question. There is a question there, many questions.
Great talk. Over here. Leonard Hayflick has criticized calorie restriction studies. He says that, I'd love to get your opinion.
Can you speak a little slowly? There's an echo here. It's just...
So Leonard Hayflick has somewhat criticized calorie restriction studies because he states that, well, you cage an animal, you feed it like an artificial calorie level, and when you start restricting the animal's calorie, you have it more similar to its wild type and like in environment studies. What would be your opinion of his criticism of calorie restriction?
I think that's a valid, totally legitimate criticism. I sort of mentioned it, and I think that's the question: is what is the right control? Because in the wild, for sure, in the evolutionary time, nutrient deprivation is the way animals live, for the most part, and so that really asks the question: is to what extent is caloric restriction extending lifespan, as opposed to that's the natural state, and when we overfeed them, we're shortening lifespan? I mean, there's no answer to that question other than saying, what is the bell-shaped curve? Where are we? And which way do you go? If you caloric restrict further, you either lengthen or shorten lifespan depending on where you are. But that's why I would say the caveat about caloric restriction is it in comparison to ad lib feeding, which it is usually, and that's, it's not just one side of the equation, it's both sides of the equation that are being compared. So it's an absolutely legitimate question and caveat, I would say, that caloric restriction uniformly extends lifespan. And in fact, there were some, two very important studies, I think. One was done in mouse, and one was done in yeast. And basically, several different groups did this, is they took, in yeast, a series of genetic knockouts in yeast, and then they calorically restricted them all the same way. Half lived longer, half lived shorter, literally half. The same result was found in mouse in 2 different studies using recombinant inbred strains of mice. Not a very powerful study, but like, I forget what it was, about 20 or 30 strains, same caloric restriction, half lived longer, half lived shorter. So, that really says you have to know where you are on that curve to know whether you're going to extend lifespan or not, and what it means to, to calorically restrict. So, thank you for the question.
There is a question here, a question there, question. Can you switch on this other microphone so that I give it?
Go ahead. Okay.
After there is this question.
First of all, thank you very much for your speak today. It was extremely interesting and very nicely introduced. I'm just starting my scientific journey. I'm in the 2nd semester of studying pharmacy. I'm potentially going to research. And I'm just asking a general question, as I have the possibility to talk to someone who has spent a lot of time contributed to aging and stem cells. As in heterochromic parabionts showed an effect of younger organisms on older. And I assume they had some sort of compatible factors that they will be able to make a pair of them, and you cannot make a pair of any species. My question is, do you think it might be possible at one point to take some stem cells of a child, for example, or a young organism, and store them in a simulating sort of mode of resilience, and reintroduce them later on into the older organism to help give treatments, for example, to damage repairs or skin repairs? Something like this is my idea. I want to know your opinion on it.
Yeah, I mean, so that kind of experiment has been done. And so, for example, transplanting a young bone marrow into an older animal and asking, does the young bone marrow rejuvenate the older animal? And in fact, so far the answer has been no. And the reason, and I think this fits with a lot of data we have, is that there is such a dominant aging environment into which these cells are placed that they become older more than the animal becomes younger. So it's kind of a thought experiment. How many cells would you have to replace in an old animal to overcome the aging environment of that old animal? How many different tissues? So it's just a thought experiment, but I think that kind of experiment is not so simple, that just giving a young stem cell will rejuvenate a tissue. It seems quite the opposite. What I didn't talk about, I didn't mention this today, I talked about this yesterday. I mentioned the heterochronic parabiosis, where the old animal some tissues appear rejuvenated. The young animal has accelerated aging. So this is not a unidirectional communication. The old animal is communicating to the young animal and suppressing functions. So there's clearly a balance in terms of, that blood exchange. Yeah.
Thank you very much. This was great. I'm a bit still thinking about this DNA damage aspect. So
the which?
The DNA damage aspect
that
occurs during aging.
Yes.
So during reproduction, clearly the DNA damage must be cleared, right? Otherwise, the whole point of reproduction would not work. But what happens when the overexpress, for example, the Ocampo story you mentioned, right? That they overexpress the Yamanaka factors.
Yes.
Will this affect also the DNA damage or not?
This is still an open question. So, even if you ask, how do egg cells age, right? Because, as an egg cell age, there has to be DNA instability and yet we reproduce as species. So, I'd be interested in other thoughts on this. This is not my specialty, but clearly there could be one of a couple things. One, there could be a selection. So the cells that have the least amount of genomic burden are, you don't like that, okay? Just let me have for an egg, for example, and even the cells that are reprogrammed by Yamanaka factors, the cells that make iPS cells might be those that are the least burdened. So that's just a possibility. The other is that, that cells, whether it's, germline cells or reprogramming cells can tolerate a certain amount of genomic instability that we just have, you know. It would enhance over generation. I mean, I don't know the answer to this question. I mean, this is a question I struggle with all the time. How does that happen? Because clearly, there's not a template for making new genomic strands. I mean, everything else in the cell is templated. You can start with DNA and make everything new. But if once you have genomic instability, you have DNA damage, other than interchromosomal, it's hard to envision what that is. But there has to be some tolerance or selection. I guess I would leave it at that and saying, other than that, I have no idea. I'd be interested in your thoughts on this, actually. Yeah.
First there.
Yeah.
Yeah, I would second your hypothesis about selection and all these. When the egg sperm production, there is a very strong selection bottleneck. Most cells don't make it. So, yeah. But I'm curious. You were talking about these experiments in rats where the rat lifespan is basically twofold longer.
Yeah, yeah.
Can you say anything? This is not really much covered in the papers. How do these rats behave during their lifetime? I mean, the beetles are basically frozen, not doing anything. Is it sort of similar thing happening to rats?
Right. I mean, one of the cardinal features of caloric restriction is that not only do they live longer, but they're healthier. And there are some studies in C. elegans, in worms, using genetic models of lifespan extension where they are not healthier. And actually, they get old and they live a long time, but as old worms. And that's not what we want. Obviously, we want healthier, older animals and humans. And for the most part, in studies in mammals, in rodents, the extension of lifespan is accompanied by an extension of health span with no obvious detriment. Now, and I think that's really been well documented. But I think that another kind of lesson that I take from what I talked about today is be careful about what the trade-offs you don't know, and that if there's a way of reprogramming a cell to become younger, so say fasting, and whoever asked this question before, if it makes a cell more resilient, that's great. But if you try and repair a tissue while you're fasting, you are not going to repair as well because you have this energy, deprivation. And so in caloric restriction, in the laboratory, where the animals are not subjected to any other kind of stress, they appear to lead healthier, longer lives. If you put them in the wild, would they be unable to compete? There may be other things that we're not testing for. And I would actually say there's always a trade-off. There's no free lunch, and that for whatever we get in lifespan extension, there's some trade-off somewhere. We may not see it, but if you look hard, I think you'll find it. So, but just back to the point, health span is extended. They get fewer diseases. So there's something definitely positive, more than just the length of life.
The last question, and after we need to close. Yeah.
Actually, regarding the caloric restriction, you talked about that it impaired the regeneration of the muscle. Is the regeneration of the muscle also impaired if you just release ketones into the mice? So if you, yeah.
I don't think we've done that, but I don't think we've done that experiment. We should have done, but I think the answer will be no. I mean, again, so by adding ketones, we're just adding another source. We're making the animals unless ketosis in itself has a negative effect on some immune cells. So actually, I mean, I shouldn't jump. I don't know the answer to it. We haven't done that experiment.
Okay.
Yeah.
Thank you.
Yeah.
You can ask questions during the coffee break. Now we have a coffee break of 30 minutes, roughly. And of course, please stay here because we will continue later on for the second part. Up. So thank you for being here again. So I'm going to open the second part of this symposium. And it's a great pleasure for me to invite on the podium Professor Marc Schulz. He's Associate Director of the Harvard Study of Adult Development and a Professor of Psychology and Director of Data Science at Bryn Mawr College. So Professor Schulz is also well known because he's an award-winning professor, a co-author, together with Robert Waldinger, of the New York Times best-selling book, The Good Life. he has directed many study and program that concern the study of social determinants that can impact on longevity. So it's a great pleasure for us to have you here and to listen from you and to learn something that probably many here do not know. Thank you.
Thank you. It's a real pleasure to be with you this afternoon. Thank you all for coming back and listening to some more. This will be a little different than the talks earlier today. I'm going to talk about a study that's been going on for over 8 decades, and I'm going to start by telling you a story about some of the people in the study. Let's see if we can master the technology. Okay, so I'm going to introduce you to John and Leo. John is on the left here. John grew up in Ohio, in a city in Ohio, Cleveland. He was from a wealthy family. His family were manufacturers of clothing and textiles. He was a good student in school, but he was bullied growing up. In those days, he was born in the 1920s. We didn't use those terms, but he talked about being teased ruthlessly as he was a kid. Because he was a good student and he was quite earnest in his studies, he got into Harvard University and went to Harvard in his teens. He was a philosophy major, decided early on not to go in the path of his family in terms of business and manufacturing. In his sophomore year, he, along with a number of other students, were asked to join the study that I'll describe in a moment. During his studies, the war broke out here in Europe, and he tried to enlist but was unable for medical reasons and served stateside during the war. So he did his duty but did it on this side of the Atlantic. After the war was over, he went to the University of Chicago for law school. He was someone that was always interested in ideas from that philosophy major, spent a lot of time thinking in his head, went to the University of Chicago, which was one of the top law schools in the United States. He graduated near the top of his class, started working for a leading firm, had a very, very successful professional life as a lawyer, worked on consultations with the government, and also taught at the University of Chicago for a long time. He got married at age 34, which was unusual for a man of his era, so it was late in life. Leo, the man on the right, came from an upper middle class family in Vermont, in New England, in the States. He also was a very good student and didn't report any of the kind of teasing that John had as a child. He dreamed of being a writer when he grew up. He too went to Harvard. He was a history major, really engaged and loved his studies while he was at Harvard, and joined the study in his sophomore year. He served in the military in the Pacific Theater. He served in the Navy. During his time in the Navy, he wrote very detailed notes of his experiences. He kept journals, hoping that he'd one day get to publish this after the war. His father died while he was serving in the war, and after the war was over, his mother developed Parkinson's disease, and he decided to move back to Vermont, was never able to pursue his dreams as a writer, actually took a job as a high school teacher, sort of on a lark, because it was something he could do when he was home with his mom, but kept teaching high school for 40 years for the rest of his career. At the same time, when he moved back home, he met the love of his life, and that was a person who remained an important part of his life for his entire lifetime. This is a study that began in the 1930s. I'll describe it in more detail, but one of the early notes about Leo was the following. I came away from our visit with Leo with the impression that the subject was, well, somewhat ordinary. So we'll see how Leo turned out. So let me describe a little bit more by giving you some data from the study. We followed them into middle age. This was when they were age 50. There were a series of questions that they responded to. The first one was, life has more pain than pleasure, to which John said true, and Leo said false. I often feel starved for affection. John said true, Leo said false. These are real responses, by the way. I'm not making up any of this. A man feels good when. John gave a somewhat tricky to figure out what he meant answer. He's able to respond to inner drives. He talked a lot about self and his needs and the ways in which he imagined he could actualize himself in real life. Leo, very different. He senses that his family loves him despite everything. A little bit later now, they're age 62. This is now 1987. When you're upset, about something that is not related to your wife, what do you do? John, I keep it to myself. I tough it out. Leo, I go talk to her, his wife. It's very natural. I certainly don't keep it to myself. She's my confidant. So one more piece of data here, 2004. This is somewhat dramatic, but we'll make the case that I'm trying to make. We interviewed participants about their intimate relationships with their partners. And we started by asking them to describe, using 5 adjectives, that relationship. John's adjectives, I should say this is John's second wife. He got divorced and remarried. Tension, distant, dismissive, intolerant, and painful. Leo, comforting, challenging, feisty, pervasive, and beautiful. So I'm going to give away the punchline before I finish the rest of my talk, but it's not the success in life, it's not your financial success, your occupational success that may be most important. There's something about relationships that's particularly powerful, and John and Leo help make that case. So I'm going to make it with some data as well. So this study, I said, started over 8 decades ago. It started in the 1930s. Interesting time. It started in Boston. It was the end of the Depression in the United States. It was also the throes of World War II. And in Boston, somehow there was a focus on human thriving. So instead of studying problems or disease outcomes, there was an interest in trying to understand flourishing. And the study enacted this in 2 very different places. It actually started as 2 different studies. So 2/3 of the original participants came from the inner city of Boston. This was Boston's poorest neighborhoods. They were disadvantaged kids that were facing quite a lot of challenges in their life. They were living in very cramped and difficult circumstances in tenement buildings that didn't have running water or toilets. Most of them came from immigrant families fleeing poverty or discrimination abroad, so they were new immigrants as well, doubly marginalized. The remaining 1/3 of the sample were in a very different place in terms of their socioeconomic position in life, but were in a different area of Boston. They were on the outskirts of Boston and Cambridge. They were students at Harvard University like John and Leo, and that was 268, or roughly 1/3 of the total sample here. All of these 724, initially all men, were followed across their entire lives in quite dramatic detail as their lives unfolded into middle life and into late life, and in fact to the end of life. Across time, we expanded the study, so the spouses or partners of the original participants became part of the study, a really important part of the study. And we're now studying the more than 1,300 children of those participants as well. This is active, ongoing research after 85 years. The data were collected in a number of ways. The questions have shifted and changed across the years, but the study has always been interested in trying to understand the daily life of its participants and also trying to get inside people's heads in a psychological way and understand how they perceive the world and experience the world. So the study began with lengthy interviews and visits to the homes of all 724 participants. Interviews were lengthy and held over multiple days with participants. From the beginning, there were medical exams and, in fact, very careful physical exams as well. There was an interest in the 1930s in the shape of people's skulls and the shape of their bodies as predictive of their personalities, ideas that haven't really held up, but were of interest at the time. Over the next 80 years or so, questionnaires about every 2 years, lengthy psychological questionnaires, medical records collected regularly every 5 years, interviews about every 10 years, and over the last 20 years or so, a number of more modern techniques like brain scanning or collecting blood assays to try and understand immune functioning, inflammatory patterns, epigenetic effects, and things like that. We also bring people into the lab and watch them go through real-life scenarios, so we stress folks out and we watch how they recover both physiologically and emotionally. We watch them in interactions with important people in their lives, including their partners as well. So that's 85 years of research. When we step back, and given the focus of today's symposia, there's some lessons that I want to especially zero in on that are relevant to our discussion today. The first one, this is a study along with many others that suggests we need to take good care of our body, that if we want to live a good life, a long life, we need to expect that our bodies are going to last for 100 years. We need to take care of it. So that means smoking is problematic, excessive drinking is problematic, Exercise is a good thing, and seeing your doctor regularly, especially as you age, is a good thing. So this is consistent with many other studies, but this is part of the findings of the 85 years from our study. Social factors matter, and that's what I'm going to talk about primarily. I'm going to briefly touch on social status, but I'm going to focus mainly on relationships, as I suggested with those two stories. So this is a slide that rather dramatically shows some of the disparities that are associated with socioeconomic differences in the sample. So the red group is the inner city men on the lower part of this graph. The purple line, it looks like it comes out purple, are the folks who were students at Harvard. We call them the college men, and I've just put a vertical line there at about age 65. This is the percentage of that sample that either had died or was disabled at that particular time. And what you see is a growing separation there as people age, but by age 65, there's a difference of about 30 percentage points. So quite dramatic socioeconomic disparities. These reflect things that are ongoing and still exist in the US. Their source is probably multifold, but this is, again, a dramatic illustration. I'll add one other piece of data. On average, the inner city men died 10 years earlier than the college men. So quite a dramatic difference between the two samples. One of the things that my collaborator, Bob Waldinger, and I have done over the last 5 years, partly in preparation for the book that we wrote, is to really try and step back. It's something that's hard to do sometimes as scientists, to try and look back and look at the headlines and see if there's an overall picture that the hundreds and hundreds of findings from the study might suggest. And when we did that, a few things stood out to us, and this is just a sampling of some of the headlines. They're all relationship headlines. If we look at midlife relationship satisfaction, that's a better predictor of late life physical health and psychological health, late life being in the 80s, than things like cholesterol. So relationships matter in a way that some of our most important risk factors, at least as important as those factors. Across 6 or 7 decades, we see a link between the experiences that our participants were having in childhood that we were able to get through interviews and observations with their parents, and their late life functioning in close relationships, and also the way they cope with adversity in midlife. So we call this the long reach, but it's clear that relationships reach across decades. In this case, 60 to 70 years that reach is present. Time spent each day, this is a finding that's quite common, but we found it over 8 days. We followed people very closely, checking in on their activities, and one of the strongest predictors of their daily mood, how happy they were each day, was how much time they spent with others that day. And finally, if we look carefully at the quality of intimate relationships late in life, this is, again, when folks are in their 80s, and then we follow them a few years later, which we can do in a prospective longitudinal study like this, we find a connection to brain health as measured by cognitive activities that measure brain health and cognitive capacities. This is in a 3-year period. It's particularly true for the women in the study, more so than the men in the study. So these are really teases just to give you a sense that there are lots of connections there. And I want to say that the connection between relationships and happiness is one of those not surprising findings. We knew that that was likely to be there. We all expect that that's true. It's really the connection between relationships and physical health that was particularly striking to us. These findings started to accumulate 20 or 30 years ago in our data and other people's data. And now, as we could see in the earlier talks from today, we're really starting to understand some of the mechanisms that might connect. Lots of experiences in life, but also relationships in particular, how they might literally get under our skin and affect our health. I'm going to not give you a detailed story about those pathways. Instead, I'm going to tell you a little story about a really interesting research project that dramatically illustrates the point that I'm trying to make. So this was research done by a husband and wife team, Janice Kiko Glazer and her husband Ron Glazer. And what they did is they gave people punch biopsies on their forearms there, or on the inside of their arm. So just a little like pencil eraser size hole in their arm. And they watched very carefully how these wounds would heal across time. And in the first study they did, they compared folks who were caretaking for people with dementia, typically their partners, with people who didn't have that caretaker burden. And the wounds of the caretakers took significantly longer to heal, on the order of 20% longer across the time. Since then, they've done a number of other studies. The one that I want to highlight is folks that are in conflictual, tension-filled intimate relationships, their wounds take longer to heal, again, on the order of people that have a caretaker burden when we compare them to folks that are in satisfying non-conflictual relationships. So this is a vivid illustration. Relationships literally figure out a way to get under our skin and influence our healing. The mechanisms are becoming more and more clear about the ways in which relationships affect our body. Certainly stress pathways are important. Relationships are pretty critical in helping us deal with stress, but it's inflammatory responses, epigenetic effects, some of the things that we've been talking about earlier today. I suggested 5 years ago, Bob and I sat down and really started to think about this, and the motive was a TED Talk that Bob had given that had attracted a fair amount of attention. And this was really our first attempt to communicate what, again, was hundreds of scientific papers to a general audience and to see whether there was an interest in that. The TED Talk went viral. We were convinced it was time to write a book. And for that book, what we tried to do was to go beyond our study to look at many other studies so that we could talk about findings that were robust across time. These were a group of men originally who were born in the 1920s. So were these studies true across cohort or across time? Was it true across gender, across culture? A number of differences that could be important. And the findings that I'm going to talk about do pass that test. It's science-based, but it's animated by the kinds of stories that I've told you already about John and Leo. And it also includes some ancient wisdom sprinkled in there, at least when that wisdom is consistent with modern science. Finally, the book integrates actionable insights, and I'm going to give you some of those ideas, at least at a cultural level, that we might think about to improve social connection. So the book has a really simple conclusion that I can summarize easily. It's good relationships that keep us happier and healthier throughout the lifespan. Some of the data that went into that conclusion I've already given you. It's all relationships, so I may have dwelled a bit more on intimate relationships just because that's a particular interest of mine. But it's all types of relationships, and I'm going to try and illustrate that for you in just a second. I'm going to show you in a second some data, which is from a meta-analysis, a study of studies about the most important risk factors for mortality. So this is for death. And you could tell me, particularly in this audience, I think we have a lot of medical students and folks from that world. You could tell me what the likely suspects are. So they're highlighted in that sort of blue color. On the bottom, I'll just start with hypertension, high blood pressure, obesity. I'm going up now to physical activity, alcohol consumption, and smoking are risk factors. The width of these bars here horizontally indicate the degree of risk. Again, this is a study of studies, so it's a reasonable indicator of the general risk associated with each of these things. So the question is what's missing here, and this particular meta-analysis added these factors. So we can measure social connection in a variety of ways, the number of friends people have, the number of support, or the quality of support that people have. It doesn't much matter how you measure it. There's a clear indicator that the absence of social connection is associated with premature death. The risk is on the order of the physical risk factors that I talked about earlier. So this is really important data. This is originally from a study that I can barely read, 2010, 13 years old. Since then, there are many, many more studies that have made this case, and we've only grown more confident in this link. We can see this probably most clearly in the interest in loneliness. In the UK in 2018, they saw this as such a problem that they made an appointment at the cabinet level for a minister of loneliness to try and conquer that. In the United States, we're catching up more recently. So in May, our Surgeon General, who has read the literature, has contributed a book in this area as well, declared that there was a crisis of social connection. And this announcement had to do both with the prevalence of loneliness or disconnection. Loneliness is a subjective feeling that people don't know you, don't care about you, that there's no one out there that really knows you. So he was concerned about the prevalence of loneliness. Most Western societies, even a number of industrialized Eastern societies, loneliness rates are somewhere between 25 and 50% of the population will report feeling lonely in a given week. So unfortunately, highly prevalent. And because of the risk that I just showed you, it really should be thought of as a public health problem. I really like the summary, so I'm going to show it to you and read it. It's really well done. What if there is something in our everyday lives that can transform our whole health and well-being? something that can decrease the risk of developing and worsening heart disease, anxiety, high blood pressure, dementia, depression, diabetes. It exists. It's something that needs to be cared for and nurtured like a garden by all of us. It can create healthier, more prosperous, and resilient communities. That something is called social connection. Quite amazing for the highest, this is the highest public health official in the United States to make a declaration about a crisis in social connection. This is a movement that's been brewing for a while in the United States, supported by research that suggests and supports this call for urgency. I'm going to make the case with data. Again, this is data from 1988, so we should have figured this out a long time ago. This was an article appeared in Science. It's, again, a study of studies. It's complicated data, but I'm going to help us go through it. On the y-axis or vertical axis, you have age-adjusted mortality rates here. It's 5 different studies with multiple samples. I'm going to just focus you on 2 of the samples here to make it easy. So on the top is a sample from rural Georgia. This is a place not of great wealth in the rural part of Georgia in the south in the United States. And they had both a black and a white sample. And what you see are blacks growing up in Evans County, Georgia, have a higher risk of death at any age than whites. The difference is not as dramatic as we might expect, but there is a clear difference. If we compare that to folks growing up in Finland, folks in Finland have a lower risk at any age of dying. Again, to translate it into longevity, much more likely to live to an older age if you grow up in eastern Finland than if you grow up in rural Georgia. What's really interesting about this, though, is not the absolute differences across samples. It's if we look at the relationship between social integration or social connection, which is measured on the x-axis, again, measured in a variety of different ways that suggest that this is a robust finding. What we see is across all of these samples, there's a general downward negative relationship between social connection and age-adjusted mortality risk. So to put it in simple words, people who are more socially connected have less risk of dying at any age. So I'm going to turn now to some of the practical importance here, and I'm going to focus mainly not at the individual level, though I'll say a few words about that, but thinking more broadly at societal, organizational levels. By the way, we use that word social fitness intentionally. So one of the most important things we can do as individuals and as a society is to recognize that social connections don't just happen. When you have as much as 50% of the population saying they're lonely, we need help, and we need to do it in a kind of effortful, proactive way. So social fitness is intended to build on a physical fitness metaphor. So what can we do? Some awareness and education things. We can work especially around loneliness to normalize the experience. When we're talking about 25 to 50% of the population, it's quite common, more common for young people and older people to experience loneliness. So we can normalize this as opposed to stigmatize this, right? Young people, at least in the States, are told that when you go to college, it should be the happiest time of your life. You should have more fun than you'll ever have in your life. These are old people who are kind of disgruntled with their life, wish they were back in college. And it turns out that there are struggles that young people at least are having these days, and one of those are struggles around disconnection. We need to do a better job of educating healthcare professionals, both in terms of the risk associated with disconnection or loneliness, and also figuring out ways to screen for this, to ask the right questions. There are screening tools out there that are widely used. It doesn't take very much time. It also has a great impact on the likelihood that people will follow up care in appropriate ways, adhere to a medical regimen. So it's worth asking questions about the networks that people have supporting them. We need to attend to vulnerable populations. So across most societies, what we see are extremes in loneliness. The extremes are the ends of the age spectrum. So the elderly are at great risk, often because of physical challenges and social isolation. Young people, it's more complicated. Young people in university are surrounded by many others, close proximity, doing similar things, but they report rates of loneliness that are quite high and quite concerning. Some of the things we can do are kind of intergenerational connection programs. Young people have things to teach older people, and without any question, older people have wisdom to share and time to share with young people as well. really think it's important. Another source of our challenge in the social connection world is that there's skills that are learnable and teachable that we know are associated with better relationships. Those are skills that relate to naming, identifying, sharing emotions. There are skills that relate to solving conflict. These are things that can be taught, best to be taught early, so they can be part of school education, starting in kindergartens and nursery schools and certainly in primary school. But they're also things that folks can engage in as adults to learn these skills and to apply them to their life. There are a bunch of kind of deconnectors out in the world, and I want to just identify them and recognize them. It's not that I want them to go away, but these are challenges that we face in modern life. The first are screens. So there are estimates in the States that people are spending more than 10 hours a day on screens. That includes the computers that folks have up and the phones that folks have up. Some of it is for work, but some of it is not for work, and people get stuck in that sort of hole of screens. So we need to just sort of temper that. Is that where we want to spend our time? Is that the best place for the things that we're trying to prioritize? What role are screens playing in our life? Jobs and work. So most of us spend a great deal of our working hours at work. I'm no different. Work can become a kind of area of consumed attention and focus for people. It's particularly true for young people who are worried about what the world may be like and what their future may be like. So we need to figure out ways to balance our work commitments and the meaning that work provides with our interest and connection. One hint, and I'm not going to talk about this, is to make sure, if we can at work, to get as much social connection out of work that we can possibly get. And then finally, stress. When I'm stressed, I'm not a good partner. I have trouble listening. This is true for most of us, that one of the skills that we need to learn to be better at relationships is to manage stress and to be able to navigate with those challenges. I want to talk a little bit about technologies that are important, and it's easy to blame phones and focus on phones as the source of the problem for lots of things, including loneliness. I want to remind people that there are new technologies introduced all the time. So when Leo and John were growing up, newspapers were introduced, and people would complain that people would get on the trolleys and the trains and bury their head in a newspaper and not talk to anyone. So that should be familiar to us when we walk into a room and people are looking at their phones, or we get on any kind of transportation. So the real trick with any technology is how do we maximize the advantages, and these new technologies are incredibly important at connecting us during the pandemic and the lockdown, essential for people who are isolated, either because they're different than others or they're remote in terms of a rural area. These technologies are really critical. They've helped work teams in numerous ways. So there are advantages that we need to try and maximize, but there are also drawbacks that we need to manage in some way. I'm just going to focus on two right now, and I'm going to start with video and technology mediation. So talking on Zoom, any kind of video mediation, any of the platforms that are out there for that use. We know from our own research, and there's other research that confirms this, there's less affect, there's less emotion, and for some people that's great. But the sad news is that less affect generally means less connection. There are also fewer signals. It's harder to pick up on people. Think about it, we're just watching their faces and their upper torso. When we were evolving as humans, we probably needed to see the whole body posture to get a sense about whether people were friend or foe. So there's some challenges associated with this video mediation. And what we find is that people feel less connected to the other person they're talking to if it's mediated via these technologies. A text to a loved one, a text to someone saying they're thinking about you, great, really terrific, but it doesn't substitute for those real in-person interactions. Social media, I'm not going to say anything incredibly insightful about this, other than we all know, and we know it increasingly so, that social media has really created a set of pernicious opportunities for social comparison. You go online, you look not just at what your neighbor has accumulated, but you can look at what the world has accumulated and what they're doing. And those social comparisons generally make people less happy. It's pretty clear. It's particularly problematic for adolescents, and it's a particularly risk factor for young women and female adolescents as well. this is a good intro for the next talk, I hope, but we want to think about designing spaces that promote connection. So the same Surgeon General that declared a crisis in social disconnection talked about improving our social infrastructure. So what does that mean? I think this is a fountain somewhere in Zurich, if I got it right. Does anyone recognize it? It's somewhere in Zurich. That's what the caption said. Places where people gather are great. Places where people gather and do things, activities together. That's how we get to know people even better. So community gardens, green spaces, libraries with activities, playgrounds where kids are coming and also parents are having opportunities to meet and be together. Terrific. We need to think hard about all of us. It's not just governments, it's educational settings, it's business settings. What can we do to promote social connection in the context that we're working? So one of the key pieces, particularly coming out of the pandemic with seeing the virtues of virtual meetings, we need to really think about balancing efficiency and social connection. So my young colleagues in particular, it's really helpful for me to be home. I have young children. I don't really want to come in for the commute. It's, it takes time out of my day. I totally get that. What we're giving up, though, are the times before meetings, the time after meetings, that time that we used to spend by the coffee machine or the cooler. And we just need to get more creative in creating those social spaces if we're going to depend on hybrid work. Really important, too, for leaders in all sectors to model for others what you mean, your priority and social connection. So Vivek Murthy, our Surgeon General, has a nice idea, which is to start every Zoom meeting with 5 minutes, allowing one person to say a little bit about what they've been up to in the last week. There are lots of creative solutions, but giving the space and energy to those social connections is really important. I've been in a lot of meetings where people skip through the introduction phase of a meeting. You don't know who you're meeting with. And then at the very end, they said, oh, I'm sorry, I didn't do introductions. So we often focus a lot on the efficiency and the transactional value, and we forget the importance of connection. Some personal steps. I suggest that we want to be proactive to our social fitness, really critical. We don't want to take it for granted. When we interviewed our participants, including John and Leo, late in life, about their regrets, the most common regret by far was something about relationships that were lost or withered across time. So we need to proactively invest in those relationships. If we're going to be successful, we need to put in the time and energy to do that. So our first task is giving our attention, which is really our most precious resource, taking it from the places that we might give it even less consciously, like screens, and to give it to the person that we're trying to bestow some affection or interest in. So showing up, making the time to do that, being present, being curious when you're present with others as well. So most of us and most people at work, when they talk about being burnt out or feeling alone, what they talk about is they really just want to be seen, heard, and understood. So if we're curious, that's the antidote for that feeling of kind of alienation that people have. This was a slide that I thought long and hard about taking out. I've done this talk now all over the States, or versions of this talk. I've talked about our research. And I've also done it across Europe. And people tell me, what you're about to say, they say it afterwards, is true maybe in the Midwest or in Chicago in the United States, but it's not true here. So let me tell you a story, and we'll see if it's true in Switzerland. Sorry, I can't go backwards. I want you to focus on the train. This was a study that was done at the University of Chicago. In Chicago, most people commute on an elevated train or they take a bus to work. These clever researchers caught people before their commute. The first thing they asked is, how are you feeling? They asked them a bunch of questions about their mood. And then they said, what do you do on your commute? And most people said, I nap, I sleep, I zone out, I listen to music, I listen to a podcast. Do you ever talk to people? No, I would never do that. Why not? People wouldn't be interested in what I say. They would reject me, is what people said. And I don't want to be intrusive either. Switzerland, listen, I don't want to be intrusive either. They caught people at clever research. They caught people at the end of their commute. First thing they said, how are you feeling? I left out one important piece. They randomly assigned half the people to talk to strangers and half to do what they usually do. They caught people at the end, how you're feeling. The people who talked to strangers, lo and behold, reported elevated mood compared to the folks who did what they usually did. This study has been replicated in other places. It's not just a Midwestern United States thing. It doesn't have to be talking to strangers. I've been on trains. I've spent a sabbatical 6 months in Switzerland. I'm a little bit familiar with this. quiet trains, don't want to be intrusive. Maybe it's not talking to strangers, but maybe it's asking your barista something other than what their name is. You know, how long have you been doing this, John? What's your favorite sports team? What tends to happen is that we get a jolt of energy when we get to know someone, either that we didn't know or we get to know something about a person that we've seen repeatedly, but we know little about. That jolt of energy isn't immediate, so as I talk about this, I get these lovely stories. You know, I had this experience yesterday. I talked to my barista. I went home and I told my boyfriend about it at night, and today I'm still excited about it. I want to tell you about exactly what that interaction is. So there's something about being reminded that there are things that are greater than yourself, particularly around social connection, that might be particularly useful. This is why I mean all relationships. The bottom picture there is from a series of ads in the United States that many people find funny. I think they're quite funny. It's for an insurance company. The man on the left is a coach who is trying to help middle-aged people who have started to act like their parents. And they do very conservative kinds of things, like worry about getting places on time and getting parking and leaving before the crowd is coming. This particular one is about talking in the elevator. So the middle guy is the client, and he just can't help but talk to strangers. So I want to share, Swiss folks in the audience, that in the United States, people are quite ambivalent about talking to strangers. There's lots of cultural mores and I think messages that you shouldn't talk to strangers. I encourage you to try it. Thank you for your interest and attention. Questions?
Question. So I realized that my mother was mistaken because she used to tell me, don't talk with strangers.
There you go.
But we need to talk with strangers. So why the mothers tell this to kids? Probably it's a problem.
It's a really important question. And again, it's not just in Switzerland that folks get this lesson. Most of us have a bias towards protecting ourselves. So when we go to parties, when we go downstairs to get coffee and cookies at the lovely reception, we worry that no one's going to talk to us or I'm going to say something silly. Probably made sense evolutionarily to worry in a kind of overly vigilant way about potential threats. But when it comes to our relationships, it's either our mothers or our heads that mess us up a lot. So we overestimate the fear. Certainly bad things can happen, so I don't want to minimize that relationships they can be very difficult. People get hurt and worse in relationships. But generally, that experience is not the case.
Colleen?
Thank you. Wonderful talk. I really enjoyed it. I'm a molecular biologist in aging, and what we do, we use epigenetics data or proteomics data or transcriptomics data to build aging clocks to predict the age of a person, and also we try to predict how long they live. Have you been thinking about using what you have in your study to build a clock for relationships?
We've certainly had those conversations, and I mentioned that we've taken blood, and the primary use of the blood so far is to get some epigenetic data. So we have some idea when we collected the data of what genes are being turned off and turned on. We have had discussions about trying to develop a kind of clock idea using that epigenetic data. We haven't done it yet. Always open to collaborations. One of the things that we've learned, we have blood, and we actually have the brains of some of the original participants. We have genetic information from that first generation. We've taken a very conservative approach in some ways, so we've frozen, for example, some of the samples with the idea that maybe in 5 to 10 years we'll have even more ideas about how to pursue it. So yeah, interested in those questions. I think that's a really important area, is trying to use biological markers of aging instead of just age. So yeah.
There is a question, sorry, first there and after the other three. Yeah.
Hello. Hi. Being alone or being in a bad relationship, what is life worse?
Which is worse? Is that the question?
Yeah, what is being alone or being in a bad relationship? Which one is worse?
Yeah, it's a really important question. I have to say, again, as a scientist, I worry about what I call horse race comparisons. It's really hard to compare those two things. And I want to say that it's not just the physical isolation that's the key here. It's a perception that other people don't have your back, that they don't know you, and it's clear that that's quite pernicious. Having said that, being in an intimate relationship that's filled with conflict, in which you feel like you're being hurt or even abused, that also has health consequences as well. I don't think we can say clearly which is worse. They're both bad.
Thank you.
There in the left.
Yeah. Hello. First of all, thank you for the talk. My question is about you. I mean, you showed how good a predictor it is, but I mean, social relationships correlate with the character, right? Which correlates with basically everything on green scored Earth, and even the like, we only have a sample size of 300 only. I mean, it's an insanely large study, but still. So what gives you confidence to say that this is not just like a hidden confounder, but actually a causality?
Yeah, so really important question here about social relationships are correlated with a lot of things, including lots of psychological variables for sure. There are a few ways that we try and parse this so that we have an idea that it could be causal. One is that we don't depend on one kind of research, so there's lots of good animal research that suggests that relationships are important. There we can manipulate rearing, for example, with mouse pups or other animals and look at the effects on folks, their behavioral signs, but also their physical signs as well. We can look at this prospectively so that we clearly establish that there's a temporal link in the direction that we're thinking about. But I also want to say that it shouldn't be surprising to us that some of these things are reciprocal, that relationships affect our mood affects our relationship abilities, and they in turn both affect our health, and health affects them as well. So I think we need to think in more complicated ways about this, but in working on this book, we started very cautiously. What could we say clearly that was responsible to the science? And I think it's like smoking and cancer. We knew long before that there was a causal connection from a preponderance of evidence across lots of research streams. And I think it's pretty clear that relationships affect our physical health in a causal way, is what I will say.
Hi, thanks for the interesting talk. It goes a little bit in a similar direction. Does it matter more the quality or the quantity? Because now in the apéro thing, I just sat with my friends. Would it have been better to talk to strangers? And then also something I was wondering, how do animals factor into this? Because there's some people who really report a decrease in loneliness when they get a pet.
Yeah, great questions. So the data are complicated. What the data suggests are that people who have more social connections generally do better. But I think the biggest impact, now I'm talking more conceptually, although there's some data to support this, if we say that 20 to 50% of the population has no one in their life that they're truly connected with, as we go from 0 to 1, 1 to 2, very dramatic changes. Certainly having more people helps when we're in a crisis. Sometimes we can't get all we need out of one person. So there's logical reasons why a larger network could be beneficial. Generally, though, it's the quality. So the key question from our study was, in the middle of the night, if you're scared or hurt or sick, do you have someone that you can call? Some people said no, even though they were married, and some people could list 10 people they would call in an emergency. So some things are more important than just the conversations that you have with a stranger. Certainly, folks who can help you deal with stress and crisis, particularly important. The question about animals is really interesting, so I always start this, tend to be funny about this. The good and the bad part of animals is that they don't talk back to us directly, right? So mostly that probably could be conjured up as good. They just, are affectionate and they give us warmth and physical comfort. But they have a tougher time helping us think more clearly about the problems that we're facing or calling our attention to something we haven't thought of. And part of the reason why relationships are linked to our health is because they're so critical for dealing with stress. So really important to me that my wife will say, Marc, you're not thinking about this the right way. You're missing something important. And dogs, and particularly cats, don't do that so well. Yeah.
Okay. If there are no further questions, I really thank you, Professor Marc Schulz, for this beautiful talk. And I'm going to introduce the next speaker, Jessica Bond, to do a proper introduction of Jessica, which is going to discuss about the role of art and cultural engagement in supporting, in decreasing mortality. I need a man of culture, so I'm pleased to introduce you Luigi Di Corato, which is the chief of the Department of Culture in Lugano, and that will introduce Jessica.
Thank you so much, Andrea. Thank you, everybody. And thank you to stay till late, so 5 o'clock. Of course, it's a great honor and a big pleasure for me personally to introduce Dr. Bourne today, because she's also a member of the World Health Organization Collaborating Center for Arts and Health at the University College in London, that daily inspire us in modeling our project in culture and health in London. So thank you to be here. Since 2020, Sylvia and I, we are working in this field, and your work is so important for us. It's more than a benchmark. Dr. Jess Bourne is a senior research fellow in statistics and epidemiology in the Institute of Epidemiology and Healthcare at the University College in London. He graduated in clinical mental health science and completed his doctorate in child and adolescent mental health in University College London. Dr. Bourne works also on the APRs project, a collaboration between UCL and the University of Florida, which aims to investigate the associations between arts and culture engagement and of course, health and well-being on the population level. So thank you to be here. The floor is yours.
So thank you for that introduction. And yeah, today I will talk about the role of arts and cultural engagement in healthy aging. So first I'll say, what are the arts and why do we care about them for older adults? I'll then talk about a couple of reviews we've done of the evidence on arts and cultural engagement and health. I'll then pick out the evidence that my team has produced on kind of 3 aspects of healthy aging, so kind of healthy aging overall, dementia and cognition, mental health and well-being, and then I'll come on to the overall association. What is the effect of arts and culture on longevity? So, what is arts and cultural engagement? So the arts, whether kind of physical or an experience, should be valued in their own right, not just as a means to an end. They should provide a kind of imaginative experience, so that might include kind of novelty, creativity, a kind of original activity, and they usually should comprise or provoke a kind of emotional response in the audience. So this can be a really wide range of activities. It could be like digital arts, visual arts, culture, heritage, through to kind of hobbies, community groups. So it could be something like joining a choir. It could also include activities like gardening or general hobbies. So a really, really broad range of activities can be thought of as arts and culture. And in the UK, we have over 1 million different venues providing arts and cultural activities. So we can see that a lot of arts and cultural activities are easily accessible. They're free or cheap. They're kind of based within the community. They can be engaged in by a kind of large proportion of the population. So they can often reach people who are not accessing healthcare. So in a national survey in the UK, they asked people how much they engaged with the arts in the last year, and 76% said they engaged with the arts in some way in the last year. 73% visited a heritage site, and 51% visited a museum or gallery at least once. So we can see that these are pretty universal activities that lots of the population are engaging in. So if we can show that these have health benefits, they have real potential to support the health of the whole population. And arts activities include a wide range of potentially beneficial active ingredients. So they often provide opportunities for social interactions, for developing new social relationships, reducing loneliness. They can incorporate physical activity, elements of kind of creative expression. They can help people to deal with stress. So there's loads and loads of different active ingredients, so opportunities for people to engage in kind of health-promoting behaviors. And in a review, we've identified over 600 different mechanisms that might link arts and cultural engagement to health outcomes. So there are kind of psychological mechanisms, biological, social, and behavioral mechanisms. So based on all of this literature, we kind of argue that arts engagement should be considered a positive health behavior, like exercise, like diet, so a behavior that we should all be incorporating in some way into our lives. So, kind of looking at the evidence base for the arts on health. Back in 2019, the World Health Organization commissioned my team to perform this review of the evidence on the impacts of arts and culture on health. And even in 2019, we found there were over 3,000 studies already on this topic. And these studies used a really broad range of methods, so from kind of intervention studies, qualitative, experimental, and overall, they showed that arts and culture can benefit people's health. And these kind of findings were grouped under two sort of main themes. So first we have prevention and promotion of health. And then secondly, we have the kind of management and treatment of existing health problems. And within those two themes, we can see evidence clustering within kind of a set of sub-themes. So within prevention and promotion, we can think about the arts and culture improving the social determinants of health, supporting child development and caregiving, as well as preventing ill health across a range of different domains, as well as encouraging other health-promoting behaviors. And then thinking about management and treatment. The arts and culture can also be used to support people with mental health problems, as well as people with acute conditions, with neurodevelopmental and neurological disorders, as well as non-communicable diseases, and supporting end-of-life care. So we can see that there is evidence across a really wide range of domains, mainly from kind of, at this point, from kind of quite small intervention studies, but suggesting that when people do arts and cultural activities, this can support their health. And we have recently built on that original review with this report published earlier this year. And this report focuses on arts, culture, and population health. So in this, we review the findings from over 70 papers that we've published since 2017. And in these papers, we show really strong evidence for a link between arts and cultural engagement and health at the population level. And the important thing here to note is that this is all epidemiological evidence. So this is all from really large cohort studies, followed longitudinally, so looking at arts and culture and its effects on subsequent health in thousands of people. And the kind of evidence we've produced so far is clustered under 4 key topics. And today I'll be focusing on healthy aging and older adults. So first, what is the kind of evidence for the role of arts and cultural engagement on healthy aging overall? We know that people are living longer now, but how can we support them to remain healthy throughout that extended lifespan? So first, I wanted to start with this study where we use quite a novel approach. So this is an outcome-wide study. So basically, this approach has been recommended over more kind of traditional designs because you can include a whole range of different outcomes. So here we have 23 different measures of health. And these are both kind of subjected, self-reported measures, as well as kind of more objective physical tests and people's kind of perceptions of their performance on those tests. So doing this, we can look at the association between engagement in creative activities. So in this study specifically, we looked at how frequently people did gardening, baking or cooking, kind of needlework or sewing, and kind of creative hobbies like arts and crafts. So we looked at the combination of how long people were spending on those different activities. And we attested the association between the frequency of that engagement and all of these different outcomes. So this approach allows us to test loads of different outcomes at the same time. So we can say that it's less susceptible to bias because it means that we can see where the kind of strongest effects are and where there might also not really be any effects. So in this study, looking at healthy aging, kind of overall, we found that more creative engagement was associated with better physical functioning, better fitness, improved sleep, and also better kind of subjective perceptions of health. So for older adults who did these creative activities more often, they kind of had particularly fewer difficulties with their activities of daily living subsequently. So this was 8 years later. They also were less likely to be needing or taking sleep medication. They had kind of better grip strength, and they also kind of perceived their balance as better. So across a whole range of different domains, we're seeing that kind of doing these sort of creative activities helps people to both be kind of more functional physically, more independent, and kind of prevent decline across a range of different outcomes. And then we wanted to go on and ask about the kind of direction of these associations. Because you might think, okay, so people are healthier, that means they're more likely to engage in these arts activities. They're more able to kind of go out and do these activities. And then kind of in the reverse direction, as I've just talked about, doing these activities can support people's health. So there's essentially a reciprocal association here with a kind of positive, self-reinforcing cycle. So we looked at this in the same sample, which is about 17,000 older adults in the US, this was. And we looked at the associations between engagement in those same kind of creative activities, so like gardening, hobbies, needlework, baking and cooking, and people's perceptions of how they were aging. So these were like the ideas that as I get older, things get more difficult, I feel more kind of useless as I age. And as we hypothesized, we found evidence for this bidirectional association. So at each time point, people who were doing more creative activities were more likely to feel more positive about how they were aging. But also longitudinally across time, when people increased their creative engagement, their perceptions of how they were aging also became more positive. And then in the opposite direction as well, people who became more positive about how they were aging over time were also more likely to increase their creative engagement. So we can see there's this bidirectional kind of loop going on where these activities can support how people are aging, and then people who are aging better are then also more likely to do these activities. And we've also looked at kind of other overall kind of indices of healthy aging, so like frailty, for example. And here, this study focused on cultural engagement. So this includes things like going to theatre, to museums and galleries, to the opera. So it's different kinds of activities to the kind of more creative ones that people might do alone or in the home. These activities are much more likely to involve going out of the house and meeting other people. And we saw in this study of older adults in the UK that people who went to the theatre, museums, galleries more frequently had a lower risk of becoming frail over the following 10 years. And that was quite a nice dose-response relationship in this graph. I don't think I can get the pointer to work. But essentially, people who did the cultural activities more frequently were less likely to become frail over time. So then thinking about another outcome in older adults of dementia. And we know this is one of the kind of largest health problems facing people as they age, that dementia is really prevalent and really hard, impossible to treat. So, in this study of older adults in the UK, we looked at the impacts of both socializing and cultural engagement on the risk of developing dementia. And here, again, cultural engagement included going to the theatre, going to galleries and museums, and kind of other forms of visiting these kinds of cultural venues. And we found, again, a really nice kind of dose-response relationship between socializing and cultural engagement and risk of dementia. So for both socializing and cultural engagement, people who did these things more often, so going up to kind of from less than once a month, going up to once or twice a week or more, they were less likely to develop dementia. And if they did develop dementia, that was going to happen later on in their life. And what I think is really interesting about this study in particular is that my team compared both socializing and cultural engagement. So socializing here was the kind of how often you met up with friends, family members, neighbors. And we found that actually, independent of this socializing, there was still an effect of cultural engagement over and above socializing on risk of dementia. And actually, that seems to be a sort of larger effect of cultural engagement on lowering risk of dementia. So there seems to be something specific about cultural engagement that can reduce people's risk of developing dementia. And we asked in more recent studies whether this is driven by the influence of arts and cultural activities on cognition. So can doing these activities improve people's cognitive performance, which then lowers their risk for dementia? So we've tested this in two separate studies. The first one with the blue graph is in the UK, and then we've recently replicated that in the US. So in both, we see that doing kind of arts and cultural activities is associated with smaller declines in memory and measures of language as well. We see for some kind of the exposures, so like visiting a gallery or museum, we see quite a nice dose-response relationship here. So basically, the more frequently you do these activities, the larger the effect on memory. That doesn't hold up across all of our outcomes, but there does seem to be a pretty consistent positive effect of doing these activities on older adults' cognition. And in our recent study in the US, we compared the effects of doing the arts and the cultural activities to the effects of physical activity on cognition. So obviously exercise is really well evidenced intervention to improve cognition. And we found that doing kind of 1 to 3 hours of arts activities a week was equivalent to doing about 1 hour of vigorous physical activity a week for its effects on cognition. So I think that shows quite promising evidence as a positive control that these activities can support cognition. Just a kind of disclaimer here that, of course, the effects in the most recent study were not entirely consistent. We think this is because there were so few people kind of engaging at the highest frequencies. So this is doing these activities for more than 3 hours a week. So we think that really we've just kind of lost power at the highest groups there. There just weren't enough people to properly test this. But this is definitely something we're going to look into further. So then, moving on to mental health and well-being. So this is perhaps the most kind of, has the largest evidence base within the arts and culture literature. It has been subject to the most research, and particularly depression. Depression being one of the most common mental health problems, particularly affecting older adults who are at increased risk of depression due to kind of other health conditions, due to social isolation, not being able to get out of the house so much. So in this first study here, I looked at a sample of older adults in the US. So this is people from the Health and Retirement Study, about 19,000 participants. And we looked at how doing a range of different kind of creative activities is associated with their risk of depression 2 years later. So we kind of compared different activity types, and we found that particularly for baking and cooking, for engaging in hobbies, and for going to social, sports, and other clubs, these activities reduce the risk of older adults reporting depression 2 years later. And we can see clubs here in orange had the most kind of consistent effect. So going to a club monthly or weekly led to about a 20% reduction in the risk of depression 2 years later. So quite large effects. The kind of other activities, doing a hobby and kind of baking or cooking, it seemed to be only when people engaged in those most frequently that they reduced their risk for depression. And then you can see for the other activities, the confidence interval all cross this null line here. So there was no evidence for kind of things like reading, writing, sewing, that they reduce the risk of depression, which we were quite surprised by. But I think this kind of ties in quite well with our previous talk, that actually these activities are much more likely to be done kind of in the home, alone, so they might not involve the kind of social element of interaction that people get from going to clubs, that kind of added social identity, potential physical activity. So that could be why we didn't find an effect of them on depression. Then thinking also about cultural engagement. So again, going to theatres, museums, galleries. We've looked at this in the UK and looked at the kind of incidence of depression. So this is new cases of depression in people who are healthy at baseline. So we take a group of older adults who have never had depression, and we look at their risk of developing depression over the following 10 years. We find, again, this dose-response relationship, that people who engage in these cultural activities more frequently are less likely to develop depression over the following 10 years. And in all of these studies I've talked about so far, we have adjusted for a range of different demographic and socioeconomic factors, also some health-related factors. But we still have kind of concerns that there are some real systematic differences between the people who are engaging in arts and cultural activities and the people who aren't engaging. And this is a kind of key problem in our field, that basically the group of older adults who do report doing these activities, particularly the ones that do them most frequently, might have they're more likely to be female, they're more likely to be of white ethnicity, they're more likely to have more social connections, to have better health overall, and to be of higher socioeconomic position across a kind of range of factors. So higher education, more wealth, more assets. So it gets really hard to work out whether these are actually causal associations. So are these arts and cultural activities having a causal impact on older adults' health after we've accounted for all of the differences between the people who are and aren't doing the activities? So this is where kind of more advanced methods for causal inference become really important. So in this study, we use one of these methods, which is called propensity score matching. And basically what we do here is we aim to match participants on a range of different demographic, socioeconomic, and health-related factors. So you take the people who are engaging in the arts, and you calculate how likely they are to do that based on all of these characteristics. And the same for the people who aren't engaging in the arts. And then what you do is you use those scores to match up people. So by doing that, you're removing the effect of all of those factors on whether people engage in arts and cultural activities. So this is a way of kind of simulating a randomized controlled trial, but using observational data, so using data from a cohort study. And we did that in this study, looking specifically at participation in a community arts group. So that could be things like a choir, a theatre group, some other kind of music group, a photography group, any kind of like community arts. And we looked at the impact of that on 6 different aspects of well-being. So this is kind of moving a bit more kind of broadly away from just mental health problems, but how people are kind of overall, they're kind of flourishing and well-being levels. So we looked at life satisfaction, the experience of positive and negative emotions, people's purpose in life, and then also these 2 measures of constraints and mastery, which is basically how well people think that they're able to achieve their goals. So in this group of older adults, we found that people who participated in a community arts group were more likely to report higher life satisfaction, higher positive affect, more purpose in life, and more mastery, so feeling more able to achieve their goals. And that was independent of all of these kind of demographic, socioeconomic, and health-related factors that influence arts engagement. We didn't find any evidence for effects on the kind of more negatively framed aspects of well-being, so negative affect or constraints. But we think that these are still like relatively large effects compared to the kind of non-negative effects on the kind of more positive outcomes. So we think that the participating in these arts groups can still enhance the positive aspects of well-being, even if it's not kind of influencing the negative elements. And again, as with the kind of perceptions of aging, these are likely to be bidirectional associations. So taking part in an arts group increases someone's life satisfaction, increases their purpose in life. Someone who then is more satisfied with their life, feels that they have more purpose, might be more motivated to go out and do these activities. So they then might be more likely to join other arts groups, to go more often, and that then has subsequent more positive impacts on their well-being. So again, it's this kind of positive, beneficial cycle that we think might be at play. And you may have noticed everything that I've talked about so far today has been based on cohorts of older adults from the US and the UK. So that's where the focus of most of our work has been so far. But in the last kind of year or so, we've been trying to broaden our scope internationally and say, are the benefits of arts engagement consistent around the world? So basically all of the evidence so far has been from kind of Western countries. For my team from the US and the UK, there's also evidence kind of around Europe. But in this study, we performed the first kind of cross-cultural comparison of the impacts of arts engagement on well-being. So we included 16 different countries who were part of 5 different longitudinal cohort studies. So you can see here there's quite a few different European countries, but also countries like Japan and the US. And in this study, we looked at the impacts of having a hobby, so particularly a creative hobby, on different elements of well-being again. And we performed the analyses kind of separately within each country. And within each country, we did what's called fixed effects analyses. So basically what we're doing is we're looking at the longitudinal change in people's arts engagement and looking at whether that is associated with the change in their well-being. So you're just looking at the change within individuals, and this means that we're automatically accounting for the differences between individuals. So by just looking within people, we're basically removing the impact of between individual differences. So things like gender, socioeconomic status, all of those factors are automatically excluded from our analyses. So across the countries, we found that the prevalence of arts engagement, so the proportion of people that reported having a creative hobby, varied really quite substantially. So it went from about 50% of the population in Spain to like 96% of the population in Denmark. Switzerland was the 3rd country with about 92% of people reporting a creative hobby. And interestingly, countries where they had higher life expectancy and higher happiness were much more likely to report having a creative hobby. And then thinking about the results, the impacts on well-being across countries. So despite the kind of quite large variations in the prevalence of having a hobby, we found that the benefits were actually really quite consistent across countries. So we performed meta-analyses here to combine all of the estimates across countries, and we found overall positive effects on depressive symptoms. So people who had a creative hobby had a kind of 0.1 standard deviation lower depressive symptoms, so small but kind of consistent effects. They were also more likely to report better self-reported health overall. They were also more likely to rate themselves as being happy, really happy in the last day. And they were finally more likely to report being satisfied with their lives. So there is pretty robust evidence across countries that participating in creative hobbies is associated with better well-being. And you might have spotted here, Switzerland is a null effect there. So actually in Switzerland, in this study, we didn't find any evidence that having a creative hobby was associated with happiness or also with depressive symptoms. So that differed to quite a lot of the other countries. We're not entirely sure why. You can see that some of the countries kind of varied, but it's overall there is a positive effect. I think the main reason driving this in Switzerland will be that it was a relatively small sample. So it was only about 1,700 people in Switzerland, compared to some of the other countries had tens of thousands of people. Overall, this meta-analysis, this paper, included over 90,000 people. So it's a really huge study on a kind of global scale, showing pretty universal benefits of these arts and cultural activities on well-being. So, given that, is arts and cultural engagement associated with premature mortality or with longevity? Well, this is the last study. I know we're getting to the end of the day, so last study I'm going to talk about today. But this was a kind of seminal paper from my team that looked at the association between engaging in cultural activities, so again, theatres, museums, going to galleries, and the risk of death. And this was in about 6,700 older adults in the UK. And what this study found is that older adults who infrequently engage in cultural activities, so by infrequently, this is literally just kind of 3 or 4 times a year. So it's really not very often. But people who even did that had a 14% lower risk of dying over the following 12 years. People who frequently engaged, so this is just kind of once a month, it's still not particularly often, had a 31% lower risk of dying over the 12 years. So these are really quite large effects we're seeing. And again, there's kind of this dose-response relationship that the more frequently you do it, the better it seems to be for you. And these associations were independent of the kind of demographic, demographic, socioeconomic, and health-related factors that I was talking about adjusting for before. But what we wanted to do in this study is kind of go beyond that and look at the role of those factors in this association. So what we did is we looked at the kind of all of the different confounding factors we included and looked at the percentage of the protective association between cultural engagement and mortality that these factors explained. So overall, all of these different factors explained 42% of the association between cultural engagement and survival. So a really large proportion of the association was due to these kind of other factors. The largest contributors in the study seemed to be older adults' cognition, other forms of kind of social, civic, hobby engagement. So this links into the importance of social relationships. Mobility and disability were also really important, as was people's wealth. So all of these factors did explain a lot of the association between cultural engagement and survival. But even after adjusting for them, these are the kind of risk of death that we find according to how often people engage in cultural activities. So there's still a large and kind of protective link of cultural engagement with longevity. And this seems to be independent of other forms of kind of socializing, other health behaviors, and other kind of risk factors for health problems. So we think that this is because of a whole range of reasons, because of the impacts of arts and cultural engagement on mental, physical, and cognitive health, because of all of the social factors, because it might provide opportunities for creative expression, for learning new skills, for problem solving. There's a whole myriad of ways that arts engagement might support older adults' health and therefore link to their survival. So, just to summarize, there is really extensive and increasing evidence that arts and cultural engagement should be considered a positive health behavior. This engagement can support older adults' health in the general population. So arts and cultural activities are not just for people who already have health problems to be delivered as interventions. They're something that we should all be doing and thinking about. There's particularly strong evidence for benefits on dementia, mental health and well-being, and longevity. And now, kind of building on this evidence, thinking about the kind of practice and policy implications, there is a lot of growing support to increase people's engagement in the community. So with initiatives like social prescribing growing in popularity, which is where healthcare professionals can refer older adults to community-based activities, so things like arts and culture, but also things like volunteering and befriending. And they can get referrals to those activities, sometimes also funding towards participating in those activities to support their health. So I think that's a really exciting kind of future direction for this field. So yeah, that's me done. Thank you.
Thank you, Jessica. Question?
Hi. I see your slides before. There's a club. You list a club as a very helpful, healthy culture activity, right? So if just go out with friends.
Sorry?
Just going out with friends, drink a beer. It's a very healthy culture activity. I mean.
So this is clubs in terms of kind of like social and kind of sports activities. It's not so much like going to a club, but which I don't know about the effects on health. I think you get the kind of social benefits for going to a club, maybe not the kind of drinking that's associated often. But this is kind of more formal, so kind of often at community centres, so activity-based clubs that are offered, or going to a sports group, these kinds of things for older adults. So it's more, yeah, kind of formal community-based activities.
All right, thank you.
A question there. There.
Thank you very much for your presentation and for the solidity of your studies, which are really amazing. I wanted to make a question, but also a bit of reflection about and wanted to ask you how you are, in your group, tend to measure those impacts. Because in my doctoral thesis, I'm working on this, on how to measure the impact of the participation to, I study theater, but cultural, active cultural participation. And what I see is that sometimes there is a difficulty in finding those tools that can be really fitting the cultural activities, because sometimes there is the tendency to use classic science tools that maybe do not give account of the complexity of the artistic experience.
Yeah.
So I wanted to ask you about which tools are you using the most, and what do you think about this necessity to adapt those kind of studies to scientific standards, but also how to preserve also the cultural and artistic experience, which I think it's really peculiar, and there is someone who says cannot be measured.
Yeah, I think that's a really good question, and it's really difficult. So I think that we've kind of got two different kind of avenues of research coming in, people using the kind of more creative methods, so often coming from a more qualitative background, and using kind of participatory arts methods to allow people to kind of engage with their kind of diverse experience and reflect on those different aspects of the arts and cultural engagement. All of the stuff I've talked about today has been based more on kind of validated scales, so kind of clinician diagnoses of dementia, kind of self-report measures of mental health, but using like widely used validated scales. So the stuff we've worked on in this kind of population approach is using the more kind of scientific tools. And we have to do that because we're using data from cohort studies where they've already collected all of these experiences. We don't have any kind of input into the measures they use, unfortunately. But I still think it's useful to get that data for providing this kind of rigorous evidence base. Because if we're thinking about kind of policymakers and funders, that at the moment, what they're kind of used to working with is these kind of more objective, obviously they're all subjective, but these more kind of scientific outcomes. So that's what they want to see in terms of providing evidence for funding these activities. But I do think there's that really kind of important crossover in the middle that you've identified between kind of understanding people's more complex experiences and these kind of scientific measures that I don't think we've managed to bridge that gap yet. I think that's something that still needs a lot more work.
Last question.
Thank you for these interesting insights. I was wondering, I know that here we have these programs that make it possible for people from lower income classes to go to museums, theater, etc., sometimes even free of charge.
Yeah.
I was wondering whether there have been studies on the effect that that had on the health and happiness already, because there you kind of get rid of the socioeconomic problems in these studies.
Yeah, I think that's really important. So I don't know any kind of specific evaluations in a kind of Swiss context, but I know that, yeah, there is evidence that if we can kind of increase engagement, then that does lead to benefits. So on that basis, we would definitely expect them to kind of have more influence. This is something that we really need to work on more, and thinking about how we can make these activities more inclusive to people from a range of different backgrounds, and therefore kind of improve the impacts on health. So in the UK, we've done some work where we've looked at the associations between these kind of arts and cultural activities and well-being across different areas with different levels of deprivation. So people in areas where it's kind of less wealthy, there are fewer resources, fewer of these activities, they actually seem to get more benefit from doing these activities. So that actually seems to be a larger effect of arts engagement on well-being in more deprived areas. So this suggests that actually these kinds of interventions might even lead to even more benefit, because often the people that might be being targeted might not be linked into healthcare, they might have lower social support, so there's kind of more potential for the arts to benefit their health.
With this, I thank you. I need to close the meeting. Thank you, Dr. Bon. And I give my conclusive remark. So on behalf of IBSA Foundation, ETH, and all of us, we really thank the speakers for coming from all the parts of the world for this very interesting meeting. We have learned a lot. And we really hope that you enjoy staying here with us in Zurich. You will come soon another time. Thank you very much.